I would request everyone to keep yourself in mute. We will be starting our program very soon.
Good.
I would request everyone to please mute yourself. We are going to start now. Good morning, everyone.
Good morning, sir.
Good morning, sir. Please, I would request everyone to please mute yourself. Rupam Das, can you please mute yourself? Thank you so much. Good morning, everyone. Good morning to our Honorable Chancellor, Sir, our Honorable Vice Chancellor, respected Dean, Sir, our Chief Guest, Guest of Honor, and all our delegates present over here. My name is Joyita Bhattacharya, Assistant Professor, School of Medical Sciences.
And I am Suraj Shah, Assistant Professor, School of Medical Sciences. It is a huge privilege for us to welcome you all to the second international conference entitled Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting Edge Innovations, that is ICTRHR-2022. The conference is sponsored by Science and Engineering Research Board, Department of Science and Technology, Government of India, and collaborated with Central Ayurveda Research Institute, Ministry of Ayush, Government of India. Our media partner is Pharma Tutor.
This conference aims to provide an excellent platform to acquire the knowledge on the recent trends in the pharmaceutical discipline through the oral, poster presentations, plenary, and keynote sessions. We are also eager to hear the enriching scientific talks of our distinguished speakers around the globe that would not only transform the event into a multidisciplinary platform for innovative ideas, but would also kindle a sense of encouragement to undertake productive research works.
Prayer works wonders. Prayer should spring from the heart. To begin this day orderly, let us first invoke the presence of the Almighty with a song. The song is being presented by the faculties of our school.
एक ओंकार सतनाम करता पुरख निर्मौ निर्वैर अकाल मूरत अजूनी सैभम गुर प्रसाद जप आध सच जुगाद सच है भी सच नानक हुसे भी सच सोचे सोच न होवे जो सोची लखवार चुप्पे चुप न होवे जे लाए लहा लखतार भुखिया भुख न उतरी जे बन्ना पुरिया पार सहसा न पालक वोहे ताइक नच लेना कि वे सच यारा हुई है कि वे कूरे टुट्टे पार हुक्म रजाई चलना नानक लिखे आना तुमी निर्मलो करो मंगलो करे मुलिनो मानमो मुछाई तुमी निर्मलो करो मंगलो करे मुलिनो मानमो मुछाई तब पूर्ण किरण दिए जाग मोर मोह कालिमा घुसाए মলিনো মর্ম মুছায়ে তুমি নির্মল করো মঙ্গল করে মলিনো মর্ম মুছায়ে লক্ষ শূন্য লক্ষ বাসনা ছুটিছে গভীর
Thank you for the heavenly aerials, for letting the goodness spark to a spiritual chain reaction with your angelic voices. Thank you, everyone.
Now it is the time where we are going to release the souvenir cum abstract book of our conference. I would request our Honorable Vice Chancellor, Sir, Chief Guest, Guest of Honors, to kindly release the souvenir book. I would also request Bitasta ma'am to kindly release the e-copy of our souvenir book. Sir, please. Bitasta ma'am, can you please show the messages from our Chancellor, Sir, Vice Chancellor, Sir? Yeah, thank you so much. Chief Guest, yeah. Thank you, Bitasta ma'am. You can kindly stop the sharing. If your action inspire others to dream more, learn more, and become more, you are a leader, says John Quincy Adams. At this juncture, I would like to attribute this proverb to our Honorable Vice Chancellor, Sir, Professor Dr. Dipendra Kumar Jha. We welcome you, sir, and I would request you to kindly address the gathering. Sir, please, over to you.
Hello. I hope I am audible.
Yes, sir.
Yes, sir.
Great. Good morning, one and all. At the onset, I would like to, welcome, all the participants, the chief guest and guest of honor, and my colleagues here from School of Medical Sciences and Department of Pharmaceutical Technology. My colleague, Dr. Sajal Jha, and his team, Dr. Parth Roy, who conceptualized this and put up this great show. I was actually briefly going through the conference book, and I was pleasantly surprised to see the kind of entries that you have got, kind of speakers, galaxy of speakers that you have been able to invite and get on the show today. It's amazing. We know things have changed, and I think the first and foremost, probably the buzzword of today's era is actually your era. Actually, everyone is looking keenly, and the pandemic situation that all of us are going through, I think, of course, pharmaceutical technology, or say pharmacy, biotechnology, those areas were anyway considered to be a great areas. But nowadays I can see there is a sudden rise in the resurgent buzz among all the students, parents, and everyone around. So certainly it's an era of, it's your era actually. But then when you become suddenly very important, your area becomes suddenly very important. It's not suddenly, but I'm just talking about today's context. I think you actually discover your challenges as well. You discover that what all you can actually do to make it better, to actually serve the society in a better way. Just if I may remind you all that, and all to the people who have who know probably a little about Adams University, we are a very young university, but the vision of the university is to become an internationally recognized institution of higher learning through excellence in education, research, and innovation. Now, in this context, may I ask you to switch off or say mute you? There is a background noise. So in this context, the second international conference with the theme Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting-Edge Innovation is actually fitting to the occasion, fitting to the vision of the university. I thank first the collaborators, the Central Ayurveda Research Institute, Ministry of Ayush, Government of India, and ACRB, Department of Science and Technology, Government of India, who are our sponsors, and also the media partner, PharmaTutor. Just I would like to, I am reminded of one thing today, that what we call innovation actually is largely misunderstood. Whenever we do research, whenever we publish papers, today also, I think after these deliberations, you will be publishing papers. But I think what we do is we tend to stop at the point of invention. Certain thing we invent, certain thing, new thing we bring through deliberations, through discussions, through our own research, experimentation. But then we tend to stop those things at the point of invention. You do invent, and then you probably go for a patent, and then after that, particularly I'm talking to my colleagues from academia, that you stop at that point. Stop at that point, and then I think we do not actually innovate. Most of the things are actually stopped at the point of invention. Point of, we publish something in the form of a paper, a highly acclaimed paper probably, or a patent, and then we stop there. I think the need of the hour is actually to go beyond that. You actually meaningfully implement those inventions, and then only you can call it innovation. Otherwise, innovation will be just a word. So I think, application-oriented inventions are the need of the hour. So this would be my first, point of view, probably you may like to dwell upon. The second thing is, industry-academia partnership. I think we have seen even during this pandemic that AstraZeneca, which was a result of Oxford University and industry coming together to form, to develop something, to come up with something which actually is saving mankind, saving all of us. So I think the real collaboration between industry and academia is the need of the hour. So two things that I would like to discuss here and bring forth here, that my colleagues and all of you get inspired through the keynote lectures, through the people, the galaxy of speakers that we have, and try to take our findings to real invention. Otherwise, it will be just a piece of paper, just we do organize events, and then that's it. So I think going beyond that, meaningfully implement those inventions is the need of the hour. And then having a close collaboration with industry is also something that we need require. Then only, we can actually justify our research, particularly done in the academic circle. So I think these two things, it came to my mind. I'm actually looking very keenly to the deliberations that is scheduled over next 2 days, today and tomorrow, and actually very happy to find that a lot of industry and speakers from both industry, academia, and research circles are going to deliver their findings, deliver their share their findings, deliver their keynote and other addresses. Thank you, Professor Castro, from professor at University in Argentina, and also scientist at National Research Council in Brazil. Dr. Maha Nasser, Faculty of Pharmacy from Ain Shams University, Egypt. Professor MNV Ravi Kumar is from University of Alabama. Professor Phak Phum from Prince of Songkhla University, Thailand. Dr. Ranjita Shegokar, Director of Pharma Business Unit in Campumed, Germany. And Dr. Madan Mohan Gupta, School of Pharmacy from University of West Indies. And I think I may have missed many, but I'm really fascinated, actually, really happy to see the galaxy of speakers today. And I'm sure the deliberations are going to be beneficial for not only for our faculty colleagues, but also for our students who are budding pharmacists. Response wise, getting more than 200 abstracts from across the world, I think is a phenomenal achievement, I would say. The acceptance rate is little over 50%. That shows that we have worked on quality. We have a certain benchmark we have set for ourselves, and I can just wish you all the best, and you continue growing, you continue making this a meaningful dialogue platform. The speakers, all the participants, I hope that it was a normal time, and we would have actually delighted to welcome you on this beautiful campus of Adams University, but then it couldn't happen this time. Maybe there is always a next time. But I'm sure that you are going to, even through these e-meetings, you are going to make your friend circle larger. You will be, particularly our students and other participants will be able to forge some really lifelong friendships, which is going to enrich them both personally as well as professionally. I hope that next 2 days, today and tomorrow, are going to be really great, and maybe a time to remember amid all these tough times, trying times. At the end, I would like to wish you all the best, wish this conference all the best, and please just stay safe and stay healthy. Thank you.
Thank you so much, sir. Thank you so much for your kind and encouraging words.
Thank you.
Today, we have among us Professor Dr. Debashish Bhattacharya, Director of Medical Education, Department of Health and Family Welfare, Government of West Bengal, as our chief guest. It is an honor for me to welcome you, sir, on behalf of School of Medical Sciences, Adams University. I would request you to kindly address the gathering. Over to you, sir.
Good morning, and I think I am audible. Sir.
Yes, sir, you are absolutely audible, sir.
It's a really pleasure to be here amongst you in this auspicious occasion, and I feel our Honorable Vice Chancellor has already started the ripples. His thought processes has really meaningful in these days, because if all of us do not join hands, we cannot really make these pharmaceuticals from the lab to the bedside and finally to be useful for the humankind. Otherwise, there is no meaning. In this regard, I want to add a little bit of things, because we usually do not pick up students which will be really become entrepreneurs later on, because we have to inculcate these habits, that what usually about the patent and all these things, startups, and then ends there. We will have to go ahead with it. And another thing regarding the joining hands with the pharmaceuticals and other industries, majority of the occasions, we think about our own benefits, not the benefit of the humankind as a whole. So in this regard, we have to think it holistically so that really we stand for the people and science really comes in the benefit. Just like in this occasion, I just like to remember you, the Lord Vishnu and Mata Lakshmi. You will see in Ananta Sajja, Lord Vishnu is lying down and Lakshmi ji is doing seva of Sri Narayana. So that means Narayana symbolizes Jnana or the knowledge, and Lakshmi ji symbolizes the wealth. So that means wealth is carrying the knowledge, so that knowledge becomes much more holistic and become useful for the whole mankind. So we will have to work together to make the knowledge become useful for all of us. And I wish and pray to the Almighty that your endeavor, this beautiful endeavor, should become useful for the mankind, and wish all success to this 2-day workshop. And I also pray that Corona dies down and the next year we will be meeting physically. I hope so. And I pray that all of you remain healthy and our endeavor together will really be beneficial for the mankind. Thank you so much. And I am really sorry because I am busy in another meeting. So please excuse me. In between, if I get a time, I will join in because there are beautiful lectures. I would love to be present and hear those if I can manage time. Thank you and wish you all the best.
Thank you so much, sir, for your time.
Thank you so much, sir, for your thought-provoking address. Yes, sir. We will surely focus on those areas. Even we focus on the innovation. We have the incubation center, so where all the faculty members or the students are involved in the research and also the patent part and the entrepreneurship. Now, it is a pleasure for all of us to have the eminent personality with us, Dr. Venkata Varaprasad Payala, Director Institute, Central Ayurveda Research Institute, as the guest of honor for today's conference. Heartiest welcome you, sir. Thank you for taking out time for all of us from your busy schedule to share your thoughts with us. Once again, I welcome you to the function, sir. I would request you to kindly address the gathering, sir.
Thank you, Surajji. Thank you so much. A very good morning to all of you. Honorable Chancellor of Adams University, Professor Samit Ray, respected Professor Dipendra Kumar Jha, Vice Chancellor, Adams University, respected Devashish Bhattacharya ji, Chief Guest of the occasion, Sri Anup Kumar Sil, Registrar, Dr. Sajal Kumar Jha, Dean, School of Medical Sciences, Convener of this program, Dr. Pardharai, Co-Convener, Dr. Tanmay Guriyaji, and Dr. Manish. My dear Assistant Directors and Research Officers of my institute, all the dignitaries, office bearers of this Adams University, professors, fellow scientists, and young researchers, ladies and gentlemen, welcome to you all. I am indeed honored and privileged to be invited today for the inaugural session of Second International Conference organized by this university. It is a matter of great pleasure that the Department of Pharmaceutical Technology, School of Medical Sciences, Adams University is organizing Second International Conference on Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting-Edge Innovations in collaboration with my institute, Central Ayurveda Research Institute, under CCRS, Ministry of Ayush, Government of India. Kolkata, which is a reputed institute under Central Council for Research in Ayurvedic Sciences. Whereas my organization, CCRS, is the premier organization in the research and development in the field of Ayurveda in the Government of India. My institute, CARI Kolkata, has rich experience in clinical research and research in allied subjects like pharmacology, pharmacognosy, chemistry, biochemistry, etc. And there are several outcomes of our research done by this institute are patented. For example, I-64, an anti-malarial drug prepared by this institute, which is widely recommended by Ministry of Ayush and CCRS during this COVID-19 pandemic. We are all happy to note that Ayurveda has become household name now globally, and all have used herbs like ashwagandha, guduchi, etc. for immune boosting to prevent COVID-19 infection. I am glad to see so many scientists, high-level experts, industry experts from around the world gather here today. Quest for health and long life are perhaps the and old as human existence, and efforts are unremitting to address the challenges and triumph over the bottlenecks across this journey. Ayurveda, the science of life, evolved as a comprehensive system healthcare systematically through scientific experimentation of high order backed by sound and reproducible evidence base and stood at the test of the time. Several strategies and roadmaps are being drawn to carry forward merits of this science so as to meet the current day health needs and mainstream its core strength alongside through research and development in this country and across the globe. The fundamental aspects of holistic system needs adequate positioning while designing clinical trials to examine the safety and efficacy of Ayurvedic approaches. Furthermore, the other challenges and issues related to quality and safety, namely dosage forms, delivery system, diverse concepts, and complex approaches in trial design, diagnosis and therapy, outcome of clinical efficacy, and drug interactions are also pose certain limitations in research. The need of the hour is to adopt a systems approach to validate the therapies and approaches with the integration of principles of Ayurveda and biomedicine without losing the vital fundamentals of both the systems. Such an approach with well-designed research plans could possibly facilitate to generate tangible evidence. The research and development in the field of oil systems in different areas such as drug development, including quality assurance, preclinical safety evaluation, and clinical research are being conducted at different levels, such as research councils under Ayush, Ministry of Ayush, academic institutions, both Ayush and non-Ayush institutes, such as medical colleges, pharma colleges, universities, etc., and other research organizations. As far as I believe, the main theme of this conference is to develop and discuss new ideas, creations, and disseminations in the field of drug discovery. And this conference will provide new insight into drug development to the industries, the academic institutions, researchers, and other stakeholders. Finally, I hope that the conference will be fruitful for all the participants. In this occasion, I welcome all the chairpersons, the eminent speakers for this conference. I would also like to congratulate everyone who made great contributions in organizing this two-day conference. I wish this two-day conference a great success and hope university will organize many more such conferences and conduct several academic activities in future too. Thanks to organizers for collaborating with my institute and looking forward to more of such activities and research collaborations with you. Thank you one and all. Thank you. On behalf of my officers and staff, I thank the organizers for inviting me today. Thank you so much. Thank you.
Thank you, sir, for your encouraging word. I hope we will be doing great things with the partnership with Ayush.
Yeah.
Like the rainwater, bringing life and prosperity to the land, your words bring enthusiasm and strength in our hearts. Let me now take the opportunity to welcome the backbone of our School of Medical Sciences, the Associate Dean, Professor Dr. Shajul Kumar Jha. Sir, we welcome you and request you to kindly address the gathering with your motivational words. Over to you, sir.
Yeah, good morning to everyone. And thank you, Joyita, for giving me this opportunity. Respected Vice Chancellor, sir, Chief Guest of today's event, Dr. Deposition Bhattacharya, the Director of Medical Education, Government of West Bengal, and the guest of honor of today's conference, Dr. Venkat Varaprasad Piala of Ayush, the international speakers, delegates, my dear colleagues, my dear students, very good morning to one and all. On behalf of School of Medical Science at Amas University, I would like to welcome you to the second international conference on, the title is the Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting Edge Innovations. And we are lucky that we got the collaboration with the Central Ayurveda Research Institutions and the funded by the Department of Science and Technology, SCRB, Government of India. And I also I would like to thank our media partner, Pharma Tutor. Now, the objective of this conference, today's conference, is to bring together all the leading academic scientists, the industrialists, and the research, all the aspects of the pharmaceutical and the healthcare field. And definitely this will offer a common platform for interaction with these eminent academicians. And there will be a great exchange of the thought process, and the lectures and seminars would be delivered by the most experienced persons in the field of research and academics. Now, as VC sir rightly said regarding innovation and invention, now in addition to that, just one more component I want to highlight in this pandemic era: lifestyle diseases. These are also, now we are everywhere, we are discussing about the lifestyle diseases. These lifestyles are actually evolving rapidly due to this evolving technologies and rapid exchange of information across the globe. And in addition to that, normally we have seen that due to this climate change, which actually accelerate the geographical dynamics of the disease. And because of that, we are getting, the result is that both communicable and non-communicable form of disease actually poses more challenges, and which we have never faced before. And the perception of the way this pharmaceutical sciences we are dealing with such changes is under precedented scrutiny. So here the innovation is very, very important, and that innovation we have to convert into invention, a tangible product, a tangible outcome, definitely which will serve the suffering humanity. So that is why the pharmaceutical innovation nowadays playing a vital role, because our target is, the pharmaceutical innovation is targeting or the main target is, we have to improve the healthy living. We have to improve the what you call the overall health, and we need to meet the expectation of the society. At the same time, we have to contribute, and whenever there is urgently required, urgently needed, and worthy effect both in the, and that is why industrial and academic researchers, we need to work together, and then only we can bring some tangible outcome, which will definitely there are challenges are there, but definitely we can overcome these challenges and will deliver a product which will definitely be useful for the suffering humanity. Because as a healthcare provider, this is our primary duties and responsibilities. So innovation in pharmaceutical sciences actually is needed to meet the current challenges, normally imposed by the societal at the same time environmental demands. So definitely I'm hoping that these 2 days actually is a perfect stage for discussion and learning, and to what you call a lot of networking, as VC has said rightly. There will be a lot of exchange of ideas, there will be a lot of networking will be there. So I wish the conference grand success, and at the end I wish to thank my management, the respected Vice Chancellor sir, who is always motivating us, Honorable Chancellor sir, and entire management of Adamas University, to providing us an opportunity to do this conference. So thank you one and all.
Thank you, sir, for the continuous motivation. Your words mean a lot to us, for they carry a lot of wisdom and vision for our future. Thank you so much, sir. We are now at the end of this inaugural function. To give a perfect end to this session, may I now request our convener, Dr. Partha Roy, Associate Professor, School of Medical Sciences, Adamas University, to deliver the words of gratitude. Over to you, sir.
Okay, thank you, Joyita. Thank you for giving me this opportunity to conclude the inaugural session of our international conference organized by School of Medical Sciences. Very good morning to one and all, and a very warm welcome to all the delegates, participants, distinguished speakers, as well as young and senior scientists, academicians who have directly or indirectly contributed in making this extravagant event happen. It would have been, I feel it would have been really, really fantastic, as our BC sir was telling, that if we are able to welcome you all physically in our lush green campus. But under the present conditions, we are forced to transform and we are forced to change and welcome you all virtually. So keeping this metamorphosis in mind, actually we designed and we entitled our conference, Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting Edge Innovations. Though this transformation that we are talking about has actually affected each and every corner of the healthcare sector. But when we think of the what are different corners of the society, the healthcare sector seems to be mostly affected, because the causative agent behind this change is actually the COVID-19 virus. I sincerely hope and pray, as our chief guest has told, that this scientific extravaganza marks the prelude to the end of the pandemic era, and we are able to come out of this virtual world and meet and work on the real life as we were doing normally before. So as today I am bestowed to give the vote of thanks, at the onset I would like to thank Almighty for keeping us all safe and also triggering our brains to conceptualize this event. As every dream of School of Medical Sciences cannot approach reality until we have the unconditional support of our Honorable Chancellor Sir and Honorable VC Sir, as always. Along with that, we need the inspirations as we get, and as well as the day-to-day feedback from our beloved Dean and HOD Sir. I would also like to express my gratitude to DST SERB Government of India for extending the funding support to the event, Central Ayurveda Research Institutes for holding our hands for this event, our media partner, Pharma Tutor, and Director ME Medical Education for gracing the session as a chief guest. Secondly, I would also like to convey my regards to all globally acclaimed stalwarts of healthcare research who have accepted our invitation to present their views, to present their research in the conference through plenary and keynote sessions. Thirdly, I would like to acknowledge all senior and young scientists and academicians who have agreed to participate and contribute in this conference. Last but not the least, I would like to pay my regards to my co-convener, Dr. Tanmay Guria, and the emblem of my pride, Team School of Medical Sciences. In fact, I do not have mere words to thank my team. Just I would like to say that when Team SOMS supports, even landing on moon seems easy. Now come, let us all sink into ICTRHR 2022 to infuse us with new hopes, concepts, and strategies that will actually enable the innovations that we make to reach from the lab to the bedside. Thank you. Over to Joyita.
Thank you so much, sir. Now we will be going for a short break, and we will be returning with our first session sharply at 12 p.m. IST. I would request everyone to join back at 12 p.m. sharply with our first session. Thank you all. I would request everyone to keep yourself on mute. We will be starting our sessions shortly. I would request everyone to keep yourself on mute.
Srija, I think our speaker, Professor Manasar, has joined.
Yes, sir.
Prof. Maha, am I audible to you?
Hello, good morning.
Yeah, good morning. Very good morning.
Very good morning, madam.
Thanks for
Good morning,
accepting
everyone.
Madam.
Thanks for accepting our invitation and presenting your work.
Thank you so much for the kind invitation.
Yeah.
Always great to be with you.
Yeah, it would have been lucky that if we were able to have you on board or in our campus again, but hopefully our next conferences will try to make you that you come over here and you deliver your talk in our campus only.
Yeah, it would be a pleasure.
Yeah. We'll start the session very soon.
Good to have you back, Professor Maha.
Hello, good morning.
Hello, this is Roger Trey. Good to have you back with us.
Thank you so much. It was an amazing experience at Adamas last time.
It could have been better if you could come personally and have spent time with us. And now
yes, next time.
Next time. Right. Okay. So, are you planning to go out of Egypt nowadays? No, you are coming out for conferences and all.
Yeah, we would be starting to go, I think, starting from March, physically, because after 2 years of COVID, we really need to travel and go outside of the country. So I'll start like next month.
Okay. Good to hear that. How is it?
Yeah,
how is
hopefully.
Egypt? COVID scenario in Egypt? How is it?
Yeah, it is. Yeah, things are stable. It's fine.
Fine. Right. I think it's time for your lecture.
It's, I think, 4 minutes left, so we'll start sharp at 12. Yeah.
A very good afternoon to all the dignitaries, guests, delegate, and our most beloved students with immense joy and pleasure. I hereby welcome you to the first technical session of the 2 days international conference. I, Srija Shur, Assistant Professor, Department of Pharmaceutical Technology, Adamus University, welcome you all to the first technical session of the 2 days international conference. Today's program includes technical talks of 4 renowned speakers. Therefore, starting off with the first technical session by our eminent resource person, Dr. Mahanasar, who is the Faculty of Pharmacy from Ain Shan University, Cairo, Egypt. She will be delivering her lecture on the topic Clinical Applications of Nanoparticle, Reality or Fiction. For conducting the session smoothly, I would like to invite our respected chairperson, Professor Rudra Prasad Shaha, who is the Associate Dean and HOD in the School of Life Science and Biotechnology, Adamus University. So now I would like to hand over the stage to Professor Shaha so that he can initiate and conduct our first technical session smoothly. Sir, please.
Thank you. Thank you, Srija. And on behalf of Adamus University, first I'd like to welcome Dr. Mahanasar for agreeing to attend the session and deliver your talk on clinical applications of nanoparticles, reality or fiction. So before you start, let me introduce yourself to the audience. Dr. Mahanasar is an associate professor and researcher of pharmaceutics and industrial pharmacy, and the executive director of the academic cooperation and international relations sector at the Faculty of Pharmacy, Ain Shams University, Egypt. She has published more than 100 international papers in the field of drug discovery and nanotechnology. Dr. Maha was awarded with AKN Regional Award in the field of life sciences in 2019, the State Encouragement Prize in the field of advanced technological sciences, serving the medical sciences 2017, Obada International Prize for Distinguished Researcher under 40 years in 2018, the ACDI IMA Egypt Award for the Best Applied Research in 2018, Comstech Award for the Best Research Paper in Biological Sciences in 2017, Professor Shauki Al-Haddad Award in the field of cancer tumors in 2018, Professor Abdo Salam Award in the field of medicinal products in 2020, and the Egyptian Excellence Accolades of the highest grade in 2020. She is also a peer reviewer in more than 100 international scientific journals and an editorial board member in 12 journals, including Nature Publishing House and Cell Press. Dr. Maha was granted several awards by diverse funding bodies such as Foundation pour University de Lyon, TWAS Biovision Unit in Bibliotheca Alexandria, FIP Foundation for Education and Research, the National Academy of Sciences, Robert Bosch Stiftung Foundation, and the National Institute of Health. She is also Africa Science Leadership Program ASLP Fellow, TWAS and African Academy of Sciences Young Affiliate, a Next Einstein Forum NEF Fellow, a member of the Global Young Academy, a member of the Egyptian Young Academy of Sciences, and she is currently an expert of the World Economic Forum Expert Network. She was also elected president of the Arab-German Academy of Sciences and Humanities. So with a short introduction, I would like to welcome Dr. Mahanasa to deliver her talk.
Thank you so much, Professor Shaha, for the introduction. Thank you so much. And I would really like to thank Professor Dr. Partha Roy and Professor Rajat Ray for the kind invitation to Adamas University. I've been in Adamas University in 2019, and it really feels like home. So I'm really happy to be here with you in this conference. So I'll start sharing my screen now. Okay, so is the presentation clear?
Yeah.
Yes, ma'am.
Yeah.
Okay, perfect. So good afternoon, everyone. My name is Dr. Maha Nasser. I'm an associate professor at the Faculty of Pharmacy in Shams University in Egypt. And today my talk is going to be about the clinical applications of nanoparticles, because my field of expertise is on the drug delivery and the use of nanoparticles, especially for preclinical and clinical applications. So today I'm going to display some of the clinical applications of nanoparticles from the research that I've been conducting. And then at the end of the session, we would answer the question if the clinical applications of nanoparticles is a reality or fiction. So nanotechnology has really benefited medicine, and there is a term which is commonly being used now, which is nanomedicine. The nanoparticles, they have the advantages of lowering the side effects of drugs, reduction of the treatment costs, and increasing the patient compliance to the medication. Of course, we all know that the nanoparticles, they can increase the bioavailability of the drugs by altering their pharmacokinetics and biodistribution, and they can allow the targeting of the nanoparticles in the desired site of action. So we have several types of nanoparticles, but basically what I prefer to work on is the biodegradable, either polymeric or lipidic nanoparticles, such as the liposomes and the vesicular derivatives. And we are going to display some of the applications of these polymeric and the lipidic particles in the current presentation. So basically, I'm going to focus on the clinical studies that we have been conducting here in Egypt and in collaboration with other countries as well, including the application of nanoparticles basically for dermatological diseases. So firstly, we are going to start with the androgenic alopecia disease, which is loss of hair, which is caused by the dihydrotestosterone. It causes hair fall in both men and women and causes premature baldness, and it's a very severe problem. And probably all the patients who are suffering from androgenic alopecia, they are using minoxidil, which is a chemical substance, and they all report suffering from side effects such as palpitations and cardiovascular side effects. And once they stop the treatment, the side effects continues, and even the hair starts to fall again. So the recent research that we have been conducting, it showed a connection between the androgenic alopecia and oxidative stress. So we thought, okay, why don't we use an antioxidant drug like melatonin, for example, and see how it can behave in androgenic alopecia. But of course, melatonin suffers from pharmaceutical limitations such as inability to penetrate the skin and inability to exert its therapeutic action unless it's inside the carrier. So we have loaded melatonin in nanoparticles, which are called nanostructured lipid carriers. As you can see in the TEM picture, it's basically formed of solid lipids and oils. And the oils that we have been using to prepare the nanostructured lipid carriers were antioxidant oils, in which the olive oil gave the most prominent results. And then the key to the therapeutic action of these topical nanoparticles is their skin deposition. So we have conducted an experiment in which we have calculated and quantified the amount of melatonin that has penetrated in the several skin layers, such as the stratum corneum, epidermis, and dermis. And we have managed to prove that the nanostructured lipid carriers were able to deposit the melatonin in high amounts in the skin. And this was reflected in the therapeutic action. As you can see in the pictures, these are actual clinical pictures from patients who are using either the melatonin solution, which is the solution of the drug in PBS. And the second picture, it shows the patients who are using the nanostructured lipid carriers. And as you can see, the increased thickness of the hair, the increased growth of the hair, and the decreased hair loss, and it's evident from the pictures. So we have proven that the loading of an antioxidant in nanoparticles, it managed to treat a disease which is stubborn like the androgenic alopecia. So we have also attempted, since we have proven that antioxidant molecules, they are beneficial for the treatment of androgenic alopecia, we have prepared nanoparticles as well, which are lipidic nanoparticles from ascorbyl palmitate, which is the ester derivative of the vitamin C. Of course, you all know that vitamin C is known for its antioxidant activity, and the lipophilic form of the vitamin C can be used as a bilayer forming molecule in order to form nanoparticles. So basically what we have done is that we have prepared the vitamin C vesicles, we have loaded melatonin in the vitamin C vesicles, and then we have checked the therapeutic action in the patients. As you can see also in the pictures, that the patients who were using the vitamin C vesicles displayed increased thickness of the hair and decreased hair loss. But if we are to compare the vitamin C particles with the nanostructured lipid carriers, that I have reported previously, the nanostructured lipid carriers were superior to the vesicles. So we said, okay, if there is a relation between oxidative stress and androgenic alopecia, why not test a very powerful antioxidant molecule, which is known as the coenzyme Q10. The coenzyme Q10 is a very potent antioxidant, and it's commonly used in the topical products. However, it's a very lipophilic molecule, and its penetration across the skin is very hard because it also has a high molecular weight. So in this work, we have prepared several phospholipidic vesicles like liposomes and other derivatives which are elastic vesicles like the transferosomes, ethosomes, transethosomes, and they are elastic vesicles that have the ability to penetrate to the skin to a deeper extent than the liposomes. And then the winning vesicles that we have deduced was the transethosomes, which is basically phospholipidic vesicles containing surfactant and ethanol in its composition. And as you can see from the table, the deposition was extremely high compared to the other vesicles. So it proceeded to the clinical trials, which was conducted on both males and females. And as you can see, after the 3rd month, there was significant improvement in the hair loss and increase in the hair growth in male and female patients. So we have deduced that using antioxidants in nanoparticles, which are biodegradable lipidic nanoparticles, can really benefit the patients suffering from androgenic alopecia. The second disease that we were concerned about was the psoriasis. And of course, you all know that the psoriasis is an inflammatory disorder. It's an autoimmune disease, and it's really troublesome for the patients. And some of the patients, as you can see in the picture, they have persistent psoriasis, which is diffuse all over the body, not even localized in a certain area. So we said, okay, why don't we test nanoparticles in patients suffering from psoriasis? And the first type of nanoparticles that I have thought of was the microemulsion, which is basically very minute nanoparticles. As you can see from the 10 pictures, we can obtain small nanoparticles even in the range of less than 100 nanometers. And the first microemulsion that we have thought about was formed from jojoba oil, which is an oil known for its anti-inflammatory activity. So we have constructed a phase diagram, and we have conducted skin deposition modeling in order to delineate the ultimate composition, which can lead to high skin deposition, but not passing to the systemic circulation. Because the topical diseases, we are focused on the deposition in the skin, and we don't want the drugs to go into the systemic circulation, because in this paper, we were using the tazarotene, which is a synthetic retinoid. So we wanted to deposit it in the skin, but it rather doesn't go to the bloodstream. So we have loaded the tazarotene in microemulsion after, of course, we have conducted skin deposition modeling, and we have tested several concentrations of surfactants, oils, and co-surfactants. And as you can see in the picture, the optimum formulation caused extreme alleviation of the symptoms, decrease in the inflammatory lesions, either in the trunk area or in the back. And it was proven very promising in the treatment of psoriasis. Then we have found that some of the patients, they really require severe treatment, not just the synthetic retinoid, and they are even prescribed methotrexate. Of course, you all know that methotrexate, it's a cytotoxic drug, and it has extremely side effects because it's a cytotoxic drug and it's used in the treatment of cancer. But it is prescribed in the severe cases of the psoriasis. So the methotrexate, when it's administered orally or it's administered like by injection, it causes severe side effects, especially to the liver. So the dermatologist came to us and said, okay, can you formulate a product or a formulation which loads the methotrexate and just deposits the drug in the skin and doesn't go to the systemic circulation? And we would be using the laser in order to enhance the penetration of the methotrexate as well. Because the methotrexate, it's a very challenging molecule topically because it's like hydrophilic molecule, and its penetration to the skin is very low. So it was really a challenge, and we loaded it in the jojoba oil microemulsion, and we have combined it with the fractional erbium laser, and we have obtained very nice results with the patients, and it was proven histologically. And when we took blood samples from the patients, the methotrexate did not go to the bloodstream, so it was really great result for us that the methotrexate was able to be concentrated in the skin, but doesn't go deeper into the blood. Another application of the psoriasis, the treatment of psoriasis, we said, okay, why not test a natural material like like extracts from olive oil, because we have noticed that patients who were consuming the olive oil in their diet or just putting olive oil on their skin, which is suffering from psoriasis, they have alleviation of the symptoms. Of course, it's not a great improvement, but that led us to the conclusion that there must be something inside the olive leaf that causes the improvement of the symptoms in psoriasis. So we have done our research, and we found that the oleuropein was the key ingredient found in the olive oil and the olive leaf, and it's responsible for the antipsoriatic activity. So for the first time, we have reported the use of the oleuropein in a microemulsion system, and we have tried it on the patients in comparison to clobetasol, which is a corticosteroid, which is commonly prescribed for the patients suffering from psoriasis. Of course, you all know that corticosteroids, they suffer from severe side effects for the patients. So we have compared the nano system that we have prepared from the oleuropein to the corticosteroid, and the results were really great. We have seen great and excellent improvement in the patients using the oleuropein microemulsion compared to those using the corticosteroid. So also that was a great result for us, and we can honestly say that revert back to nature, stop the corticosteroids, because nanotechnology can help deliver the natural material across the skin. In this paper, we have also deduced that the patients who are suffering from psoriasis, they also suffer from depletion of the ceramide lipid. The ceramide lipid, it's commonly found in the skin of normal patients, but in the patients who are suffering, from psoriasis, they have a depletion of the ceramide. And the depletion of this ceramide, it causes the skin to be dry, causes the skin to be inflammatory, and it increases the water loss from the skin. So we said, okay, why don't we prepare nanoparticles which are supplemented with the ceramide lipid. And from this idea, we have developed serosomes. And serosomes, they are vesicles. They are not spherical vesicles. They are rather tubular vesicles, like the fibers, because the critical packing parameter of the ceramide is higher than the phospholipid. So it allows the vesicles to be formed in the form of tubules rather than spherical vesicles. So we have applied these serosomes on the patients, and we have noticed retention in the water in the skin and decreased inflammatory disorders. And when we loaded the serosomes with the synthetic retinoid, the tazarotene, it really led to alleviation and improvement of the psoriatic conditions in the patients. So we can also conclude that supplementing some functional additives like the ceramide lipid to the vesicles can also help in the treatment of the psoriasis. Then afterwards, we have tested elastic vesicles, which are called spanelastics, which are formed of the span surfactant as well as a penetration enhancer oil, such as the cineol. And we have loaded tazarotene as well in the elastic nanovesicles. And we have concluded that they can also lead to significant improvement in the psoriatic conditions in the patients. So we can conclude that whatever nanoparticles you are using, it's like the microemulsions, the phospholipidic vesicles, the surfactant vesicles, and the elastic vesicles, they all lead to improvement in the psoriatic conditions. And when we compare them to the marketed products, for example, market as gel, as cream, also the corticosteroids, when we always compare our nanoparticles to the products which are found in the market, they are much more superior and much better. So moving on to another disease, which is skin cancer. The skin cancer, it's somehow common in Egypt, and especially the basal cell carcinoma. And the treatment modality that is being used is the photodynamic therapy, because let me give you a brief on the photodynamic therapy. It's a mode of treatment which utilizes a light in addition to a photosensitizer. When this photosensitizer molecule, it's simply like a drug that is being activated by light at a certain wavelength. When this photosensitizer is activated by light, it causes the release of singlet oxygen that destroys the problematic area in the skin, like the warts, like the lesions, like the skin cancer areas. So usually the skin cancer it's being treated with the photodynamic therapy. However, there's a problem in the photosensitizers, the dyes like the methylene blue, the indocyanine green, for example, is that they are unstable when they are in solution. For example, in this particular research, the indocyanine green, we have found that it's very unstable and it's degraded in solution. So the photodynamic therapy using indocyanine green solution is not effective. So we thought, okay, why don't we load the indocyanine green in nanoparticles, which are also biodegradable vesicular nanoparticles, as you can see from the transmission electron microscopy picture. These are the spherical vesicles loaded with the indocyanine green. And then we apply this dispersion in addition to the photodynamic therapy to the skin of the patients. As you can see as a representative picture from the patient suffering from skin cancer, the basal cell carcinoma, we can see the alleviation of the lesion after the treatment, and it was also proven histologically. So this, when we compared the photodynamic therapy alone with the topical, with the colloidal indocyanine green and the indocyanine green, which was loaded in nanoparticles, we have 80% clearance rate in the patients. Like from every 100 patients, we have 80 patients which are significantly cured by the nanoparticle in conjunction with the photodynamic therapy. And so we can conclude that combining nanomedicine with the photodynamic therapy, it leads to really improvement of the results compared to the photodynamic therapy alone. Okay, moving on to other diseases, which are the skin fungal and viral infections. So we had a question, can the topical fungal infections, and especially the deep-seated fungal infections, or the viral infections which are resistant, like the warts, for example. Can we use the nanoparticles to treat such infections? So we have conducted several clinical trials. The first one was on the skin fungal infections, the patients suffering from the tinea. And interestingly, what we have done in this research is that we have loaded the antifungal drug, which is sertaconazole, in vesicles, and we compared the efficacy to a marketed product. And then we even challenged ourselves. We placed 20 times lesser dose of the sertaconazole in our vesicles, and we compared it with the conventional marketed cream, the Dermofix cream, and we said, okay, let's test it on the patients. And interestingly, the sertaconazole in our vesicles led to much better improvements in the patients with the nanoparticles compared to the conventional cream, even at 20 times lesser dose. This really makes us conclude that by using nanoparticles, we can really decrease the dose of the drug and decrease the side effects of the drug consequently. And it can even be reflected on the decreased expenses in the production, because the active ingredients, they are really the most expensive part in the formulation. And so if we manage to decrease the dose, it really reflects on the cost of the product. In another fungal disease, which is the candidiasis and dermatophytosis, this is still unpublished data that I just would like to share with you. We have loaded eugenol, and the eugenol is known to be a natural oil, but when you apply it on the skin, despite having antifungal activity, the application of eugenol on the skin, it causes skin irritation. So it cannot be used as such. So we loaded eugenol in the vitamin C derived nanoparticles, and we have managed to obtain high deposition of the eugenol in the skin, in the several skin layers. And when we compare the antifungal activity of the aspasomal formulations to the eugenol solution, we have seen better association of the nanoparticles with the fungal membrane, that we have even proved with the transmission microscopy. We have seen the vesicles bind with the membrane of the fungi and penetrating it, which is reflected on higher antifungal activity of the aspasomes. And as you can see in the infants suffering from candidiasis before the treatment, after treatment with our aspasomal formulations, it caused alleviation and improvement of the symptoms. And also in the patients who are suffering from dermatophytosis, it led to significant removal of the fungal lesions. So we can delineate that, yes, nanoparticles can be used for the treatment of the skin fungal infections, but what about viral infections? Can they also be treated with nanoparticles? So in this research, we have managed to load methylene blue, which is also a dye, in transferosomes, and we conducted photodynamic treatment on patients suffering from resistant plantar warts. As you all know, the plantar warts, they are viral infection and they are very painful, especially if they are in areas like the legs, and they allow the patients, they cause the patient so much pain and difficulty in walking. And so the conventional treatment for the plantar warts is the photodynamic therapy. But as I mentioned before, the photodynamic therapy, it relies on photosensitizers, and these photosensitizers, they do not penetrate the skin to a great extent. So in this work, we have loaded methylene blue in transferosomes, which are vesicular particles, which are elastic, and they can penetrate the skin to a greater extent. And when we compared the patients who were treated with photodynamic therapy just with the methylene blue solution to those who are using methylene blue solution in transferosomes, interestingly, we found that the patients who were treated with the nanoparticles, they were cured after 2 sessions in comparison with 4, 5, and 6 sessions using the conventional therapy. So this made us conclude that also using nanoparticles of the photosensitizer, in addition to the photodynamic therapy, can reduce the number of sessions that the patients have to endure, and it leads to better clinical results than the conventional photodynamic therapy. So that was another disease that we have proven nanoparticles to be promising in. And then we move to vitiligo, which is also a very challenging disease to be treated. Why? Because the patients suffering from vitiligo, they have depigmented areas, like large white areas in the skin, and it's very resistant to the treatment as well, and it requires long course of treatment. So reviewing the literature, we have found that the use of narrowband UVB light, it can stimulate the melanocytes. For example, as you can see in this picture, that's a patient suffering from vitiligo in the knees. And the vitiligo is in depigmented areas in the skin that needs melanin pigment so that it can. So using narrowband UV light can help pigment these areas, these vitiligo areas. However, it's not effective. It's not that much effective in the patients, and many patients, they declare that they see no improvement and they stop the treatment. So we thought, okay, why not use a photosensitizer? And this photosensitizer, it can be like the bergamot oil. It has a very nice smell, and it has some photosensitizing molecules like the bergaptan and bergamotin in very small amounts inside the bergamot oil, which are known to stimulate the melanocytes. So we thought, okay, why not combine with the narrowband UV light? Why not combine something which is activated by this UV light? And we have chosen this bergamot oil, and we use the bergamot oil alone with the narrowband UV lights in the patients with vitiligo. It was very irritant, and the patients said that they cannot continue with the treatment. So we loaded it in nanospanelastics, which are nanoparticles that are elastic enough to penetrate the skin, and the results were just amazing. The patients were saying that the nanospanelastics was giving them like promising results, even in the resistant areas of the treatment. I can recall a testimonial from a patient who was saying that I've been trying to treat my vitiligo for 10 years, but I haven't noticed significant and promising results until I used your product. So we were really happy with the results, and we can honestly declare that using a photosensitizer, in addition to the narrow band UV therapy, and this photosensitizer loaded in nanoparticles, it can lead to very promising results in the patients of vitiligo. It led to stimulation of the melanocytes, and immediately the depigmented areas become pigmented. We also managed to load the bergamot oil in other type of nanoparticles, which are nanostructured lipidic carriers, and we also have tested the improvement in the patients. As we can see here, when we used just the narrowband UV light, it gave moderate improvement, but when we used the bergamot oil in addition to the narrow UV band light, it gave significant improvement in the pigmentation areas, and the patients are now continuously using the product in order to allow the pigmentation of all the vitiligo areas. So that was another promising application of the nanoparticles in a dermatological disease, which is vitiligo. So what about the acne, which is a problematic condition, especially in the teenagers? And the conventional chemical treatment, it damages the skin. So we said, okay, why don't we load natural nanoparticles? And in this paper, we have used the nicotinamide, which is a vitamin B. And we have loaded it in biodegradable chitosan nanoparticles, and their structure is, as you can see here from the transmission microscope. So we had patients suffering from acne, and we have treated them for the very first time with chitosan nanoparticles. We were the first to report the clinical application of chitosan nanoparticles, especially in dermatology. And we have loaded this nicotinamide vitamin B in the chitosan nanoparticles, and we have noticed significant improvement in the acne lesions and decrease in the inflammation in the patients using the nanoparticles. So we were happy with the result. Compounds, not just vitamins, but a molecule like the quercetin. It's a natural molecule, which is a polyphenolic compound, which is derived from several vegetables and fruits. And we have loaded the quercetin in our very unique aspasomal vesicles, because it's a vitamin C derived vesicles. And these vesicles, of course, you all know that the vitamin C has anti-inflammatory properties. So loading the quercetin in the aspasomes, it also led to alleviation of the acne lesions compared to the other side, which was not treated with anything. It was just placebo. So we had significant improvement in the acne lesions when the patients used our quercetin vesicles. And then we said, okay, why don't we transform these findings into a product that can be actually used by the patients? So we thought that why not prepare patches for the skin, because we have noticed that the patients were saying that we need to cover the lesions till they heal. So we said, okay, why don't we create patches for the skin that can be placed and reside on the skin with this quercetin aspasomal particles, but we will include these aspasomal particles inside nanofibers, and we can produce these nanofibers in the form of patches that can be placed on the skin. So what we have done is that we have loaded the vesicles inside nanofibers. That's why we stated that these nanofibers are composite, because we have loaded vesicles inside the nanofibers, and we have tested them also in the patients. And as you can see, also alleviation of the acne lesions in the patients, and they were very happy with this patch because they can just go out with the patch on the lesions. And the results was really great in terms of antimicrobial assays, and we have also conducted safety assays on fibroblast cells, and the patients were really happy with the results. Then we were approached by dermatologists who were saying, we usually treat the acne using pulsed dye laser and also with the photodynamic therapy. So we need to do a comparative study to compare the efficacy of the pulsed dye laser alone with the photodynamic therapy and the nanoparticles. They said, we have heard so much about the nano products that you are developing for the photodynamic therapy, and we really want to make use of them in comparative study to the pulsed dye laser. So basically in this paper, we have compared the efficacy of the pulsed dye laser to the photodynamic therapy of the methylene blue loaded in nano-emul gel. And we compared the reduction of the acne severity between the two treatment modalities. And as you can see, the red line, it shows the PDL, which is the pulsed dye laser, and the decrease in the acne severity, it was reaching 51%. But when we calculated the decrease in the acne severity with the photodynamic therapy, in addition to the nano-emul gel, it was much greater decrease in the severity of the acne lesions. So this is also another great finding that we have proven, is that the photodynamic therapy with the nanoparticles, it's much more efficacious than the pulsed dye laser when it's used alone for the treatment of acne. Finally, we have also attempted the application of the nanoparticles in patients with melasma. And in contrast to the vitiligo, the patients of vitiligo, they have depigmented areas. In case of the melasma, the patients, they have hyperpigmented areas. As you can see in the skin, it's full of melanin and it's hyperpigmented. So the patients were saying, okay, we are using the whitening agents, but they are not of any use, the conventional gels, the conventional creams, And we need you to prepare nanoparticles from the skin whitening agent. So we have prepared chitosan nanoparticles. We have loaded them with skin whitening agent. And we have made a split face study in which part of the face, the right part, was being gels of the face was treated just with the conventional marketed gel. And the results were amazing. We have proven clinically that just the loading of the skin whitening agent in nanovesicles, it leads to removal of the hyperpigmented areas. And we have proven that also histologically when we counted the melanin pigmented areas and the active melanocytes, they were much decreased in the patients who were treated with our nano formulation. So we can also declare that nanoparticles can be beneficial in the treatment of melasma. So that we have done. But what about the current status of the nanoparticles? How far have we reached with the nanoparticles commercially and in the market and in the clinical trials? Unfortunately, upon surveying the clinicaltrials.gov, we have noticed that there are less than 10 clinical trials who have been reported to examine the therapeutic efficacy of the nanoparticles, and they were mainly on the silver nanoparticles. And when we checked the market also for the topical nanocarrier products, we have found that most of the nanocarriers, they are just cosmetic based for nourishing, for moisturizing, but not for actually treatment of skin disorders. So we can conclude that despite the preclinical and the clinical benefits of the nanoparticles, they are still in the developmental phase. And I believe that it's our role to bring these developments and these techniques to the market, because as scientists, we have a role to translate these researches and these products and these innovations into products which are actually in the market. So to conclude, we can say that nanotechnology, it's a multidisciplinary field and it shows great promise in the clinical application, especially in the dermatological diseases. And I hope that you all agree with me in this conclusion by seeing the results of our research in the clinical trials. And with that, I would like to thank my partners in research in different countries and in Egypt as well. And I would like to thank you for your hospitality. And these are actually pictures from when I was at Adamas University at the International Conference of Current Trends in Higher Education in 2019. And I'd like to thank the staff members, the students, and everyone at Adamas University. And thank you, Dr. Shaha, for the hospitality and for inviting me, and thank you, Dr. Partharoy and Dr. Raja Trey. Thank you so much, and I would like to open the floor for questions from the audience.
Thank you very much, Dr. Nasser, for your wonderful talk on clinical applications of nanoparticles, and a very interesting talk. And you have shown that decisively that when nanocarriers are used, the efficacy of the natural products or proven drugs can be enhanced, basically by targeted delivery. And it's unfortunate that most of these findings are not translated into products that the common man can use. But I hope that in future, this would be useful for everyone. It would be available for everyone to use. Thank you again for your wonderful talk. So now the session is open for questions. So if you have any question, please unmute yourself and please ask.
Thank you, Dr. Maha, for the very interesting speech. Just I want to ask you something, that you are showing the clinical data of different types of nanoparticles in psoriasis, and these particles that I also believe that these are very interesting and very we can formulate it very easily in the lab. But as Dr. Shah has also pointed out, these are not transformed into the market. They don't go to the market, they end in the lab. So can you tell us something regarding the scale up of these kind of nanoparticles, how we should approach these things? So that we don't stop at the lab scale, at least we go to the pilot scale or something, so that we can at least attract some investors or some industry people if they want to transform it into a marketed product or something, because it's showing very good, interesting data for your preclinical studies.
Thank you, Dr. Partha, for your question. It's a really important question. As you have mentioned, I have tried several types of nanoparticles, but then the type of nanoparticles which is easily scaled up in the production phase, and we are now trying to do that in lipid vesicles and surveying the products which are already now in the market, which are really cosmetics based, I have seen that the vesicular formulations, they are dominant. So I said, okay, it's probably that they are the easiest to scale up. And when I really performed the scaling up of these vesicular nanoparticles, they were really scaled up. We have now reached in collaboration with the pharmaceutical industry, scaling up of up to 10 kilograms of nanoparticles. And then we are trying to increase and scale it up more, not on the pilot, but also on the production phase. But it's a start. We have now scaled up the nanoparticles, the liposomes in particular, from the lab scale to the pilot scale on the level of 10 kilograms. And then we are trying to scale it up a bit further. So my advice would be to start with the vesicular nanoparticles because they are the most scalable of all. Hopefully we can scale up the other particles as well, but for the time being, the vesicular systems are the most scalable of all.
Good afternoon, ma'am. I'm Shrija Sur from Adamas University. Currently, I'm working as an assistant professor here. So I have one question that currently I'm working, I have completed my master's, and since my master's, I have been working on carbon dots as a nanoparticle. So it is a photosensitizer as well, and currently you can use that as a drug delivery system. So you have told a lot about combining a photosensitizer with a nanoparticle and then using it for the treatment. So can I just, I just want to know one thing, that whether if we can use carbon dot directly as a photosensitizer and also as a drug delivery system for the treatment of these diseases. And carbon dots, if we derive from phytochemical sources, they are comparatively they have a very low toxicity. They have a very low toxicity, and also they are biocompatible. So what is your take on that?
I believe it's a very, very nice idea because you will not find so many applications of the carbon dots clinically in the treatment of dermatological diseases by photosensitization. So I believe it's a really important point which is worthy of investigation. The carbon dots on its own, but then if you are going to load like photosensitizers, I believe that there is a bit of chemistry involved because you need to conjugate the photosensitizer like chemically to the carbon dots, because I've done just a little work on the carbon nanoparticles, and the physical association of a drug to the carbon nanoparticles, it didn't give us high yields. So I believe that if you are going to load like drugs, you need to chemically conjugate the like the photosensitizing molecule or drugs or whatever. But it's really interesting idea, and I'd be happy to collaborate if you want. It would be an honor.
Sure. Actually, the carbon dot itself, there are some reports which have shown that carbon dot itself is also can act as a photosensitizer. We do not, sometimes it is not important to load separately a photosensitizer. So it can also, so if we can investigate on that, it will be, though the investigation is still very low, it has been not been investigated much on the field of drug delivery, but I think it can be a good idea.
Yes, definitely. I agree.
Thank you.
Thank you. Any other questions?
Good afternoon, ma'am. This is Amartya Sahu from BFARM, Adamas University. As you were discussing about the more or less about the dermatological diseases and the application of nanoparticles on that, I just want to know that the acne, whenever we are using some cosmetic products, we used to see that the acne, the problem of acne goes away, but some time or some days or some months later, it again comes up. So is there any permanent cure in that case of acne? Because you were discussing about the patches and all that. So I want to know the permanent cure of acne or something.
Well, thank you for your question. The problem with acne is that it's linked to several causes. For example, it can be hormonal causes, it can be stress induced. So unless you stop the hormonal, like the hormonal like changes and also the stress in the life, so there will not be a permanent solution for acne. So what we can basically do is that whenever there's an acne flare and lesions that come up, we just treat it topically. But other than that, to permanently treat acne, I think it's not possible because it's linked to several causes that you cannot control.
Thank you so much, ma'am.
You're welcome.
Any other question?
Hello, ma'am. I am Irfanul from BPharm 4th year. Ma'am, I have a doubt that if it has been scaled up to then will it be available to the developing countries? The nanoparticles which are being used for dermal use.
Thank you for your question. And Egypt is a developing country. I mean, we are doing the scaling up in Egypt, and it's a developing country. And the nice thing about the vesicles is that they can be prepared from really cheap materials, not expensive. So if it's cheap and cost effective, then it can definitely be used in developing countries.
Thank you.
Welcome.
Prof, just one short question. Do you think that as you are telling about the vesicular nanoparticles, so do you think the lipid ones are better for scale up than polymeric ones in terms of stability and cost effectiveness?
Yes. Well, also basically cost-wise, the lipidic, they are cheaper than the polymeric. And we had trouble in the scaling up of the chitosan nanoparticles. So that's why we are more focused now on the lipidic particles. And even it's even found in the market that most of the commercially used nanoparticles for cancer, for example, they are lipidic based. So this also gives us an indication that they are easier to be scaled up compared to the polymeric particles.
Thank you. Thank you.
You're welcome.
Dr. Nasser, I have a short question. I was wondering, the topical cream that we use, they basically stay active for maybe for a few months to a year. So I was wondering how long these nanoparticle based the solution or emulsions that will be active for how long? Have you checked?
Well, for the academic purposes, we have checked the physical stability. But then in terms of the scaling up with the pharmaceutical companies, we have reached some formulations that are stable up to 3 years. Because we are using additives like preservatives, we're adding also antioxidants, stuff like that, which converts the formulation to an actual product. So it can be stable on shelf up to 3 years. Some formulations that are stable to 3 years.
Okay, thank you. Any other question? I guess no more questions are available. So I'd like to thank you again for your wonderful talk and the discussion. Thank you very much, Dr. Nasser.
Thank you so much. And I'm looking forward to seeing you physically in the next conference.
Okay. Sure. Thank you. Thank you.
Thank you so much.
Thank you. Thank you.
So truly, it was a huge privilege for us to listen to the insightful talk of Dr. Mahanasar. Now, before ending the session, I would like to gift a small token of gratitude from our behalf. So I would like to present a small token of gratitude for our eminent speaker, Dr. Mahanasar, from the behalf of our department. I would request Vitasta ma'am to please share the token of gratitude in the screen. Ma'am, please.
Thank you so much. It's an honor and a privilege to be with you, and looking forward to seeing you all in India next time.
Thank you. Thank you so much, ma'am.
Thank you.
Now, I would like to present a small token of gratitude for our respected chairperson, sir. Sir, please allow us to share the certificate in the screen. I would request Bitasta ma'am to please share the certificate. Ma'am, please.
Thank you. Thank you very much to all the organizers and Dr. Shajal and Dr. Rajat and Dr. Path. Thank you very much, everyone involved.
So we have come to the end of our first technical session, that is technical session 1. Therefore, with the permission of the chairperson, sir, I would like to declare this session as end.
Yes, please. Thank you.
Okay, so before ending this session, I would like to announce that we are having a lunch session for half an hour from 1 to 1:30 p.m. Post lunch, we would be having our oral presentations followed by the technical session 2. We will start our oral presentation sharp from 1:30 p.m. Therefore, I would request everyone to join back to the conference at 1:30 p.m.
Saptarshi, are you there? Saptarshi, are you there? Saptarshi, can you hear me? Saptarshi, am I audible to you?
Hello.
Yeah.
Yeah.
Am I audible?
Yes, 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 you are, sir.
Perfect. Okay. Okay, ladies and gentlemen, good afternoon, ladies and gentlemen who are present over here. So after a very graceful inaugural session today in the morning and very attractive lecture delivered by Dr. Muhammad Nazar from Egypt. So now we'll be beginning with our technical oral presentation session. That will be the first oral presentation session today. We'll be starting with that shortly. So I am Ashim Haldar, welcoming once again all the participants in our second international conference, ICTRHR-2022, sponsored by DST, SCRB, and in collaboration with CCRAS, Ministry of Ayush. So in this session, we'll be having 8 presenters who will be presenting, showcasing their research work in oral presentation format. So now to judge them, to judge the presentation, 2 experts have joined with us. They have already joined. First, I would like to introduce Dr. Sarpananda Mukherjee, sir. So he has already joined with us. Sir,
thank you.
Thank you. Welcome, sir. Sir is associated with NSHM College of Pharmaceutical Technology, Kolkata, and Dr. Momita Das Kirtania, Madam, Associate Professor, School of Medical Science. So both will be your judge. They will be evaluating your presentation, and they have vast experience on drug delivery, formulation, product design, all those areas and the field. So Before starting with the presentation directly, I would like to inform you, please go through the guidelines that already provided to you, the guidelines and the condition for presenting the oral presentation. Please adhere and stick to the time limit specified for the oral presentation. I'm repeating once again, you will be having 7 minutes for the presentation and there will be another 3 minutes for questions and answer session. So you have to complete within 7 minutes whatever content you want to deliver to the judges. So please stick to that time, otherwise you will be disqualified. So your presentation will be evaluated by the expert based on the presentation skill, time management, novelty of your work, rationality of your work, importance or significance of your work, and how you have responded to the questions asked by the expert. So now, with the permissions of our two evaluators, sir and madam, shall we start with the first presenter right now?
Yes, yes, you can proceed, no problem.
Thank you. Thank you. So I will be calling the first presenter for the oral presentation, Mr. R. Rajagopalan. Please start your presentation quickly. Mr. R. Rajagopalan. Mr. Gopalan, are you there?
Sir, I do not see anyone on with that name yet.
Okay, then we should proceed to the next one. So next, from Rajasthan, Sudipta Roy. Sudipta Roy, are you there? Please respond. Sudipta Roy, please respond if you are there. Okay. Okay, then we'll go directly to the third presenter, that is Mr. Muniraj Bhattacharya from JIS University, Kolkata. Mr. Muniraj Bhattacharya.
Yes, sir.
Yeah. So, Mr. Bhattacharya, you can start your presentation.
Sir, now only I can start?
Yes, please share your screen and start your presentation.
Okay, sir. Sir, please give me 1 minute, sir. 1 minute, sir. Sir, am I audible?
Yeah.
Yes, you are audible.
Okay. Sir, is my PPT visible, sir?
Yes, it's perfectly okay. You can go ahead.
Okay. Good afternoon, one and all present here. I'm Muniraj Bhattacharya from JIS University from the Department of Pharmaceutics. So I will be presenting my project work, which I am recently working on. That's development and characterization of first dissolving oral films containing metformins for faster onset of action. Now, why I have chosen metformin is it is a drug that is effective in treating anti-hyperglycemic condition It falls under the biguanide class and comes in the BCS class 3 section, which states that it's having poor bioavailability due to its low permeability. It is highly soluble, but it's low permeable. That's why this problem is there. So for that reason, that's the limitation that needs to be overcome through this project. So as I stated, that's my main aim or objective of the project, to overcome the low permeability problem of metformin and to produce the films and to study their efficacy. Now for coming to the materials and methodology portion, I have used HPMC, that is the hydroxypropyl methyl cellulose, and along with that polyvinyl alcohol, PVA. Those two are my polymers, and metformin itself that I stated was my API, and propylene glycol, that was the plasticizer that was used. The methodology used for producing the films is shown diagrammatically below. That is the solvent casting method, and the compositions, various compositions of polymers that I have used is also shown here. And you can see that along with overcoming the limitation, I'm also focusing on the rate of drug release depending on HPMC and PVA compositions, various compositions. Now, coming to the pre-formulation studies that I have gone through, one is the UV assay, or which I have done of my drug sample. And the next thing was the compatibility of the API and the components, the excipients that I have used. And I came through that there's no new package getting produced, and those are compatible, obviously. Now, post-production, the post-formulation studies, I have primarily done the FTIR studies of the film. As you can see, I came to notice that the films were having metformins, which were not there were no interactions present, and microscopic and normal eye images of the films are shown on the right-hand side of the screen. Now coming to some more post-formulation studies, that I have done: the thickness, weight variation, holding endurance, moisture loss, tensile strength, and surface pH. And I have seen that successfully F3 batch, and approximately all the batches, mostly and mainly the F3 batch is my most optimized batch that I have seen. And now the swelling degree, or the swelling degree percentage that I have studied and have received those datas. The drug content and disintegration times were also studied and were found to be as required. Now coming to the drug release study, USP type 2 apparatus was used for performing the drug release studies, and you can see that the drug release patterns are shown through the graph. And 233.4 nanometer was the wavelength at which UV spectrophotometric assay was done of the samples collected at time intervals. Now, when the drug release data I have received, I went through for determining the drug release kinetics, and I came to see that the Kushmir papers is being followed mainly. And that's why the Kushmir papers graph is shown on the left-hand side, and on the right-hand side, the all the R-square values that I received from the other patterns of the methods of kinetics that I studied are given on the right-hand side. You can see the Kushmir papers is being followed. And recently, I have gone through the ex vivo model study using goat skin collected from fresh goat skin that is collected from tongue. Fresh goat tongue skin from the upper layer skin of the tongue is used, which is collected from the nearly located butcher shop. And the drug release study was comparative drug release study from of API and the F3 batch, that is the most optimized batch is done to see the drug release and the drug permeation through the upper layer skin of the goat tongue was seen to be successful, as we came to see that the drug permeation was more in case of the F3 batch compared to API. Now, when I coming to the conclusion, I can say that all the characterizations came to the conclusion that the doses from F3 was the most optimized and successful, and all the data were recorded by between perfect ranges, and the more amount of HPMC there was in any dosage form, the more will be the release of the drug compared to PBA. This was the thing that I came to notice. So here are the references of the articles and journals that I used as my references. Thank you. You may ask any question if there's a.
Thank you, Mr. Bhattacharya. Now this session is open for question and answer session. Over to ma'am and sir.
Yeah,
Yes, sir.
Mr. Muniraj, it's good.
Yes, sir.
And you have given a nice presentations.
Yes,
so
sir.
I have few queries.
Yes,
First,
sir.
you just tell me why you have chosen this metformin drug, because it's already established in the market and it will have many kind of formulations.
Yes, sir. So in that case, I would like to say you that metformin is a very famous drug nowadays, and diabetes, type 2 diabetes, that is diabetes mellitus, is very much common in throughout the humans nowadays, and the rate of people infected by not infected by they are having the disease of diabetes is getting more and more day by day. So that's why, and as you were saying, in the market available dosage forms of metformins are mainly sustained release tablets. But those in that case, there's a thing that the drug availability is lesser compared to other drugs that can be delivered through that method. So as I said, that BCS class 3 is the drugs placing in the BCS classification. So for that reason, for the quicker onset of action, I chose the drug and chose the FDA dosage form.
Because a lot of metformin preparation is available as a sustained release or control release in the market. But
yes,
what
sir.
you have prepared, that is a pill. And
Sir,
you are getting most,
that's because
most
for
of the amount
faster onset of action.
Because of faster onset of action?
Yes, sir.
That is most important for a diabetic patient?
So for my film is mainly focusing on two things. That one thing I can say that drinking having a tablet at times when it's a part of our daily life, it becomes a hindrance at some time. And I came to see that it's easier to have those films.
No,
and
it's not curable actually. Okay, you have to maintain the blood glucose sugar level for a long time.
So for that reason,
until
also,
and unless if you will not focus on onset of action and if you will focus on duration of action, then it will be good for your study.
Yes, sir.
Because whatever I have seen, most of the amount is released within 14 hours. So
yes,
after
sir.
14 hours or in day, if the patients will take 2 times of your film, then
yes,
where is the novelty? You should have some novelty in the work. So you have to compare with the novelty actually. Okay, now second question is, how you have performed the IR study of your formulations? Because it's a film, correct?
Yes, sir.
So how you have done that?
For that reason, I have used for the IR study, the Bruker is the brand of that. The ATR is present. For the ATR, I have used the film to place it under the
Means
ATR.
directly you have find out the IR, correct?
Yes, sir. And for the mixtures and when in the pre-formulation section, I have produced pellets, and the transmission part of the Bruker IR machine was used.
So during your pre-formulations, did you make the plate for IR study?
Yes, sir. Pellets. Yes, sir. I have gone
And
through
for
pellets.
your formulations, you have done directly through ATR.
The ATR. Yes, sir.
So is there any validation difference between that? Because procedure is
No,
quite
sir.
different.
The validation difference will not be there, as because we know that we are using KBR for making the pellets, and blank KBR pellets must be used for the background scanning and nullifying the KBR's presence. And that's why when the KBR pellets having the
Yes,
materials of the samples,
because that is required. In
Yes, sir.
pre-formulations, you had KBR with your physical mixture.
Yes, sir. I did
But
that, sir.
because when you are doing IR of your formulations, there's
sir.
no KBR actually. So might have
Yes,
some
sir.
interactions during your pre-formulation study.
So no, there was no interactions. I came to see because KBR was nullified using the background scanning previously.
Okay, my next question: what is the application procedure?
The application procedure I have tried to show here through a diagram that the films will be placed directly over the tongue, and that's why when there will be saliva turnover, as we know, the films will be getting disintegrated and the drug will be released immediately, and the drug absorption will be through the highly vasculated layers, vasculated layers in our oral cavity. And
So it's through oral route.
Yes, sir.
Correct. Then why
Yes,
you
sir.
have performed your release study in 7.4 pH?
So for 7.4 pH, I was studying the release as because the buffer that is suggested in the pharmacopoeias for studying metformin is 7.4. In that case,
But that is not
gives
for
that,
film. That is for like any kind of parenteral formulations or where it's going directly to the circulations. But you are making
it says
film
that
and
the
you are
maximum absorbance will be there, and that's why I use the 7.4 pH. And also, sir, in our oral pH ranges between 6.5 to 8. So that's why.
But that you have not shown in your study. You have shown only in 7.4.
Yes, sir. The study is going till now, so I am to do it in the slightly acidic, that's 6.5. Also, I'm thinking of doing the studies.
And my last question: why you have taken HPMC?
Okay, so the HPMC, as I said, that hydroxypropyl methyl cellulose, it is a polymer that is easily soluble in water and has good solubility, and that's why I have chosen it for the production of the films. As we know that our saliva contains a high amount of water, that's why HPMC will help to release the drug immediately with quick disintegration also.
And last, what is the full form of BCS?
Sir, biopharmaceutical classifications.
Then what is S? BCS.
BCS classification, sir.
I am asking full form of BCS. You have mentioned B and C, not S.
Yes, sir. Sorry.
Enough from my side. Okay.
Yes, sir.
Dr. Momita, you can ask now.
Yes, sir has given very thorough suggestions. I hope if you apply it in your further research work, it would help you. Now,
Yes,
have you done any stability studies regarding your formulation?
How
stable is your formulation? What is the commercial application of this?
Ma'am, for the stability study, I'm planning to next go for the accelerated stability study. That's due. That has to be done. That is in my coming plans also. That has not been done yet, ma'am.
Okay, and do you think that the formulation you have developed, it is suitable for commercial application?
Yes, ma'am, I think so. It's suitable for commercial application, as because it's
so how can you defend your answer?
Yes. I think it will be much more it will be having much more patient compliance if compared to tablets and taking tablets again and again compared to insulin injections, and that's why I think it will be much more patient compliance that will be giving it a marketing advantage over injections and tablets.
For how long do you intend to keep that film over the tongue? What is the time duration?
Yes, ma'am. Ma'am, for the time duration, I would like to say, as you can see, the disintegration time, the maximum disintegration time that I have seen is 66 seconds approximately. But for the F3 batch that I have seen to be the most optimized, approximately 42 seconds is enough for the film to get disintegrated, and there will be no residence of the film after 1 minute over the tongue.
Okay, no more questions.
Thank you. Thank you, sir. Ma'am, Muniraj, please stop your sharing option. Stop sharing. Okay, so next presenter will be Mrs. Akila Elias. Akila Elias from Bangalore, RR College of Pharmacy. Mrs. Akila, please start your presentation.
Yes, sir. Yes, sir. I will. Very good afternoon, everyone. Today I am going myself, Mrs. Akila, Research Department of Pharmacognosy from RR College of Pharmacy, Bangalore. Oral presentation on the topic of preparation and characterization of gold nanoparticles using phenolic rich fractions of Buchanan lens. Here in our pharmaceutical field, nowadays all department is focusing on the nanoscience.
Some interruptions.
Sir?
Yes, any issue?
Sir, I am joining from the other one account. It's okay, sir. I can present.
Yes, I can see your presentation. It's on, no issue. You can go ahead. Make it full screen and go ahead.
My presentation is on preparation and characterization of gold nanoparticles using phenolic rich fractions of Buchanan lens. Today, our pharmacy field, our pharmaceutical field is mainly focusing on the nanoscience and nanotechnology. As we know what
Mrs. Akila, can you hear me? Are you there? Yes.
I think she is getting frequently disconnected.
Yeah. I think we should move on because there is a limitation in time. Oh, I have already started once again. Mrs. Akila, madam, you can see your screen. Please go ahead. You are not audible.
whether my voice is audible, sir?
Yeah, now it's coming. Please quickly start your presentation once again. Yeah.
So, today I am going to speak about the metallic nanoparticles. Here, metallic nanoparticles we can prepare by physical, chemical, or biological method. If you choose some other chemical method, means it will produce unwanted side effects. So for that, we have chosen the plant-mediated metallic nanoparticles. Here, there are number of metals available in for the therapeutics. Among that, gold is having good therapeutic efficacy. So we have chosen gold nanoparticles for the present study. So what is the objectives of my studies? First, we are collecting the particular plant which is having the capacity to reduce the gold ions into the gold nanoparticles form. Then the plant has to be authenticated. Then pharmacognostical evaluation of the particular plant has been done. And for that plant, we are doing the phenolic content determination after preparing the extract. Then from that, we are choosing the extract is showing the total phenolic, more total phenolic content. Using that extract, we are preparing the phenolic rich fraction. Using that phenolic rich fraction of the particular extract, we are preparing the gold nanoparticles by means of the green method. After that, nanoparticles has been characterized by UV, FTIR, SEM, TEM, and XRD analysis method. Here, methodology part. plant here I am using is the Buchanania lanson. Leaf portion I have taken for the study. This plant has been collected from Tamil Nadu, has been authenticated by Dr. Chelladurai, and this drug has been subjected to the morphological evaluation. The drug will be in green in color, which is having the characteristic odor, and it is having bitter taste. And this drug, once morphological evaluation, after the morphological evaluation, the drug has been subjected to the shade drying. After drying, it has been converted into the coarse powder. That coarse powder has been subjected phytochemical recording in progress. After the physicochemical and phytochemical analysis, the plant has been subjected to the extraction process using various solvents based on their polarity. Based on their polarity, like petroleum ether, chloroform, ethanol, and water. And after the preparation of this extract, total phenolic content has been determined for all kind of 4 extracts. After determining the total phenolic content determination, among the extract, which one is having good phenolic content, that has been chosen for the phenolic rich fraction preparation. So phenolic rich fraction has been prepared by dissolving the ethanolic extract into the water. Then it has been fractionated with the equal volume of the petroleum ether as well as ethyl acetate. We know ethyl acetate will solubilize most of the polar components. So phenolic component also one kind of the polar constant. So we are extracting the ethyl acetate extract, which is subjected to the evaporation process. So we are giving the name, it is the phenolic rich fraction of the ethanolic extract of the Buchanania lanson. So this phenolic rich fraction of the particular plant we are using for gold nanoparticles. Why we are using here as a reducing agent means one is it's a cheaper method. Another one is plant itself, it's having some kind of the secondary metabolite. That secondary metabolite is having the capacity to convert the ions into the metal form. So which will be in the nano size. Another advantage for using the plant constant is whenever we are subjecting or we are using plant as a reducing agent, the plant constant will go and coat on the metals, whatever we are preparing. So along with that metals, the plant constants also will produce the proper therapeutic efficacy. That means it will increase the effect of the metal, whatever we are preparing. So it will produce the synergistic effect in the pharmacological activity. For that purpose only, we are doing this metal nanoparticle synthesis by using the plant extract. So in my study, gold nanoparticles has been prepared using the phenolic rich fraction of the Buchanania lanson. Here I am taking 10 ml of the phenolic rich fraction and I am treating it with 90 ml of the solution of the chloroauric acid, which will be in the ions form. After adding this phenolic rich fraction into the chloroauric acid solution, immediately it will undergo the reduction process. And within 5 to 10 minutes, we can able to see the color change. That means light green color change into the ruby red color, which indicates the formation of the gold nanoparticles. So this confirmation we can able to assume within 5 to 10 minutes or maximum of 1 hour. So once the color change has been happened, means we are just stirring the content using the magnetic stirrer to produce the nanoparticle sedimentation. After that, we are keeping the suspension in a cool and dark place. Then we are centrifuging the solution at 10,000 RPM for 15 minutes. Then we can able to separate the gold nanoparticles from the solution. Once the gold nanoparticles has been prepared, means we are subjecting to the characterization process. So characterization, how we are doing means, one, whether the nanoparticle has been formed in the solution or not, that we are checking by means of the UV analysis. Second method, what is the shape as well as size of the nanoparticles formed,
Hello.
that we are, yes.
Madam,
sir,
sorry
yes,
to
sir.
interrupt. You have to complete within 1 minute. You have 1 minute time. Please wrap up your presentation.
So the confirmation of my result is the plant is available in the green color, which is having the characteristic odor, and that code has subjected to the physicochemical analysis that indicates the total water soluble ash, acid insoluble ash, all the extractive values within the limit, and it is subjected to the chemical screening, which indicates the presence of all kind of the secondary metabolite. And it is subjected to the total phenolic content determination. Ethanolic extract is showing more phenolic content. So that extract has chosen for the phenolic rich fraction preparation. Using that phenolic rich fraction, we have synthesized the nanoparticles. Here you can able to see the color change. The normal chloroauric acid solution has been converted into ruby red color that indicates the presence of gold nanoparticles. And again, we are confirming the formation of gold nanoparticles by means of the UV analysis. Here we are getting the peak in the range of 550 nanometer, which indicates the formation of gold nanoparticles, and which has been subjected to the XRD analysis to find out whatever formed nanoparticles, it is available in the crystalline nature or not. Here, from the peaks of the XRD analysis, we are confirming.
Excuse me, ma'am, please stop your presentation. Time is over. Now I'll directly go to the evaluator. Sir, if you have any queries, please ask.
Whatever you have explained, the total explain was on most of the things was on isolations and characterizations of that extract part. So actually you have prepared gold nanoparticles, correct?
Yes, sir.
So you just tell me what procedures you have followed. You have not mentioned any kind of procedures. You have mentioned only reactions method.
Sir, here, sir, we are choosing the plant extract, which is having more phenolic content. That extract we are choosing for preparation of the gold nanoparticles.
That is okay. I am asking what procedures you have followed for preparations of nanoparticles. In which method you have prepared nanoparticles?
So this is biological method of metal nanoparticle preparation, sir. Here we are using 10 ml of the plant extract, and we are allowing it to react with the chloroauric acid solution. Here chloroauric acid will contain the ions form of the gold.
And
it's
whatever
not present in the form of metals.
Whatever formulations you are getting, that is a suspension?
No, sir, it is a solution.
Solutions.
Liquid form. Yes, sir.
So then how we have performed the particle size determinations?
The particle size determination by means of the SEM and TEM analysis, sir.
If it is a solutions, then how come you will determine?
No, sir. Actually, we are adding the extract to the chloroacid solution. It will
then
convert
what will be
into
your
the metal form. After
what
that,
is
we are
the
going for the drying process also, sir.
Which
we are
drying?
evaporating all the liquid content. Finally, we are getting only the powder form of the gold nanoparticles.
So which dryer you have followed, or which drying method you have followed?
Sir, I have followed a hot air oven 60 degrees Celsius I maintained, sir.
You are sure about your study?
And we can go with the freeze drying.
No, you are sure that you will be able to use the hot air oven at 60-degree temperature? Because if it is a lyophilization or freeze drying, then the method is quite okay. But if you are using the hot air oven, then I'll have some doubts. And do you have any data
Actually,
on
what I did know, sir.
And you have done your preparations only for one formulations?
Yes, sir.
And where is the polydispersity index? Do you have any data about polydispersity index? You are telling that you have got particle size around 550 nanometer.
That is
correct?
UV analysis, yes, sir.
But how you are getting particle size 550 nanometer from UV? Because for particle size analysis, you should not use the UV.
Nanometer range, the solution is showing the
Then which instrument you have used for
Here.
particle size? For particle size determinations, which instrument you have used?
Same analysis, scanning electron microscopy.
By scanning electron
SEM
microscopy?
and TEM.
Same, and what is the full form
And
of
TEM.
TEM? What is the full form of TEM?
Transmission electron microscopy, sir.
So you don't have any zeta potential value, correct?
No, sir, I didn't do that.
Then you should not conclude about the surface charge actually. If you are not having any zeta potential value, then it will have many type of questions actually which can arise. Do you have any in vitro study?
Here, study I am not included. Actually,
Then
studies and all,
where is
checking the therapeutic efficacy.
Then where is the rationality of your work? Can you tell me the rationality of your work?
Here we are going with the green synthesis.
And
if you are preparing,
just go for the previous slide. In the graph, you have mentioned only nanometer. Go for the previous slide. No, not go previous. Slide number 10. Yes, here see in the graph you have mentioned nanometer. What is that? In y-axis you have given absorbance. In x-axis it is nanometer. That is particle size analysis, correct?
Analysis. Actually found in the solution. We are subjecting to the UV spectroscopy study. If the solution is giving peak in the range of 520 to 570 nanometer, means the solution is having the gold nanoparticles.
And that comparisons you have not shown here. Okay. This is from my side. I have
UV
lot of
analysis only for confirmation.
Okay.
Okay, this is from my side. Yep.
Okay. Which drug you have chosen for encapsulating in the nanoparticles? Why nanoparticles? Why not any other dosage form?
Actually, my focusing, if you are choosing nanoparticles only getting popular in the field, instead of going with the chemical method, if you are going with the plant synthesizing, we'll get two benefit. One is it is cheaper. Another one is plant itself, it will go on coat metals. So get the increased pharmacological activity, what we are discussing. Actually, my work is anti-cancer activity. Gold is having the capacity to, that means it will produce the therapeutic efficacy on cancer. So if I am choosing the plant which is already proven for the anti-cancer study, and I am using that plant for the synthesis of gold nanoparticles, means that constant, again, it will coat with the metals and it will limit the anti-cancer effect of the metals.
You have used the alcoholic fraction. Okay. You have used the alcoholic fraction.
Yes,
so if the organic solvent remains in the formulation, what may be the consequences?
Correct.
You are telling that you are using the green synthesis, but you are using the alcoholic fraction.
But washing it with water several times after the, when we are doing the filtration, the centrifugation, we are doing filtration, primary metal taking by means of the filtration process.
Okay, but is there any study which tells? Is there any study which tells that what is the amount of the organic solvent that is remaining in your formulation?
No, ma'am.
Are you sure?
Only constraints how whether it is. I didn't do, ma'am.
Okay. Thank you. No more questions.
Because
that we are doing.
If you are targeting for cancer cell, then 550 nanometer is not enough. That will not pass through the cell.
That is only for confirmation of gold nanoparticles.
But you are telling many things.
Whether the solution is having the gold nanoparticle,
Okay,
that
no
we can
problem.
confirm.
No. Thank you,
At that 550.
Okay, thank you, Mrs. Akila, madam. Madam, please stop your sharing, so that next presenter can start. Next, Miss.
Hello. Hello.
Yes.
Hello. Sir, sorry for the interference. I am my code number is number 1, but I am unfortunately I am joined
Okay,
another
fine.
system. Yeah.
I understood the fact. Yeah. Please wait for a while, then we'll call you.
Okay, sir. Okay, thank you.
Now, please, Miss Sudipta Roy, please start your presentation.
Yes, sir.
No, fine.
Sir, first there is need to stop the screen first.
Yeah, Miss Akila, madam. Yes. Now it's fine. Please go ahead.
Yes, sir. Good afternoon to my colleague and respected sirs and my fellows. Myself, Sudipta Roy, research scholar, Bhopal Noble University, Udaipur, Rajasthan, presenting development and evaluation of Carbopol 940 blank gel-based drug delivery system in pre-formulation step of project entitled to develop and evaluate antioxidant gel containing neem bark extract. My guide name is Dr. Anirudh Singh Devra. This is the contents of the study: abstract, introduction, objective, materials and methods, results, conclusions, acknowledgement, references. This is the abstract of the study. Total 12 batches of developed Carbopol 940 blank gel bases, and this gel bases were evaluated. Gels were studied by organoleptic properties, spreadability, pH viscosity, and in vitro permeation, and the kinetic release study was done on basis of the in vitro permeation data, zero-order Higuchi cross-smear papers, Kopcha, Macquarie, Banneker model. So after completing the study, formulation scored F2, F3, F5, F6, F7, F8, F9, F10, F11, F12 had been found zero-order release, whose R-square value was from 0.9299 to 0.988. In cross-smear papers model, all developed gels were predicted non-ficant super case 2 transport on basis of n exponential value greater than 0.89. In Kopcha model, ratio of A and B for formulation scored F1, F2, F3, F4, F5, F8, F9, F10, F11 were greater than 1, had been assumed control release diffusion. Developed gels were predicted control release diffusion at zero-order rate. This is the introduction of the study. This research work was based on development of Carbopol 940 blank gel base. It was developed for preparing the developing gel using neem bark extract, and to evaluate the gels through the skin layers to socialize in low cost for better therapy in predicting drug delivery system. This is the introduction. It is the objective of the study to predict drug delivery system or Carbopol 940 blank gel base for developing antioxidant gel containing neem bark extract to predict release kinetics of polymeric blank gel base of Carbopol 940. This is the fabricated diffusion cells where I have done my permeation study for this blank gel basis. This is the fabricated spreadability study apparatus. This is the blank gel Carbopol 940 polymer gel, which is shown here transparent. This is the materials and method of the study. Chemicals required: Carbopol 940 polymer, distilled water, sodium hydroxide. Carbopol 940 polymer was procured from Assis Chemical Works, Jodhpur. Apparatus requirement: water, the apparatus, osmolar viscometer for viscosity study, diesel pH meter for pH, and other instruments, microprocessor, UV. This instrument was used for the UV analysis. The blank gel bases were analyzed at 287 nanometer, which is predetermined lambda maximum in water, taking absorbance for in vitro permeation study using microprocessor UV visible double beam spectrophotometer L1270. Development of gel, blank carbapol 940 gel bases were prepared by soaking and swelling method to keep definite weight of carbapol 940 in water for 24 hours at room temperature by neutralizing with percentage sodium hydroxide solution as neutralizing agent to pH of skin 5.5. If the skin is not within the if the pH is not within the skin pH range, then the diffusion process may be delayed. For this reason, the pH was adjusted to the skin pH. So here in this literature formulation chart for the formulations code F1 to F12 is presenting here. Here in this formulation chart, carbapol 940% in gel was used as from 0.206 to 1.43%, and water was used as 99.79% to 98.56%, and sodium hydroxide 1% solution was used as quantity sufficient. This is the zero order release of formulation code F1 to F12. For zero order release equations, K0 is equal to Q2 by T. Q2 is amount drug release in milligram at time T in hour. This is a carb and graph data of amount of drug release in milligram versus time in hour. And from this graph data, it was predicted the control release diffusion. It was this formulated gels where followed the diffusion release. This is the percentage permeated graph data, this data was evaluated from the in vitro permeation study, and this is the percentage cumulative permeated data. This data is also evaluated from the was collected from the in vitro permeation study, and from this study we have taken the data was collected, and the result is here. This is the result on basis of the evaluation study color and clarity. There is a transparent gel was evaluated. There is no odor of this particular gel basis, and the gel was very much washable with the water when it was applied onto the dorsal surface of the left hand skin retention test. There is a skin retention test, and after doing the skin retention test of this particular gel basis, there is no skin retention, edema, swelling, and skin color changes. These are the results, linearity results of the percentage permeated data and percentage cumulative permeated data, and the linearity has shown for percentage permeated and percentage cumulative. This is the R square value, regression value, and the sum square residual value. And so on the basis of data, we can it can be said that this gel was confirming the zero order release. This is the different kinds of release data. This is the Higuchi model for formulation score from F1 to F12. Higuchi equation is Qt is equal to Kh. This curve was plotted between the cumulative permeated Qt versus root square of time. And from this data, it was compared with the y is equal to mx plus c, this linear equation. And from this data, Kh, Higuchi constant was calculated, R square value was calculated, SSR value, sum square residual value was calculated. This is the Kopcha model. Kopcha model was confirming.
Last 1 minute, you have to complete.
Okay, sir. This is the macroid model for formulation score. F1 to F12. This is the Crossmere-Peppers model. So these are the model which has confirmed the control release diffusion. This is the data of the zero order release, Higuchi release, and Crossmere-Peppers model, Kopcha model. This is the macroid model result. This is the result of macroid-Banneker model, and this is the result of pH. pH was within the pH, and viscosity was shown very good. This is the conclusion on basis of R square value and SSR value. From percentage permeated and percentage cumulative permeated data, formulations code F6, F7, F9, F11 were release based. Blank carbapol 940 gel base was predicted diffusion as zero order release and control release from Kopcha model. Absorption was predicted super case 2 transport as N value was obtained from more than 0.89 in Crossmere-Peppers model. On basis of SSR value, Crossmere-Peppers model, Kopcha model, and macroid-Banneker model was shown best fit because the linearity was shown very good. Was a very small number of SSR value was found from this calculations. This is the acknowledgement, and these are the references. I'm acknowledging to my guide, Dr. Anuj Singh Deora, and we are very much thankful to Dr. Amitabh Mandal and his research scholar, Department of Chemistry. Thank you, sir.
Okay, thank you. Thank you so much. Over to Tarun, please.
Why you have prepared?
Hello, sir.
Yeah.
Yes.
So sorry for the interruption.
Yeah.
Actually, according to the time, 2:25 mine is there. So can you
Yes, ma'am, I will call you. Please be there. I will call you. there was a delay little bit. Will be there. Okay. Sir, please go ahead.
Okay, Ms. Sudeepa,
Yes,
just tell me why you have prepared the gel apart from other formulations. Why you have choosing to prepare the gel of your extract?
I have my future plan is to prepare the Nimba extract, to prepare the antioxidant gel containing Nimba extract. So for this reason, I want to make the antioxidant gel in very cheap cost, because I have an object to wound healing assay. I will I will do the wound healing assay using the antioxidant gel, and gel is very low cost, and I will want to do available this Topical 940 gel for all the populations for our country, because this is very much costly process. We know that this is very costly in surgery and to cure the patient, and there is lots of medicine out there, and there is also the side effects are also there. I have chosen Nimbak extract, and Nimbak extract was taken from the plant, but particle gel is a hydrogel. It was very much controlled. It will give the very good effect when we use the plant product, because Nimbak extract.
I am asking you only one questions, and you are telling me around 10-15 answers.
Yes, sir. Yes, sir. Sorry, sir.
Carbopol itself is having wound healing property.
Yes, sir. Yes, sir.
And it's reported. Then what is the use to add neem bark extract with this carbopol? And how you will confirm that you will have synergistic effect of your formulations?
Can you repeat the question?
Carbopol itself is having wound healing property, correct?
Yes, yes.
Then neem also it's having wound healing property. So both the things is there in your formulations. So you are expecting a synergistic effect in your formulations, correct?
Yes, sir.
So how you will confirm that?
Sir, how I will confirm, sir? For this reason, I will do the self-formulation study, sir.
Because how you will be sure that this antioxidant effect or wound healing effect is because of neem?
I have, sir, I will do the self-formulation study, cell line study. I will see the cell formation, neem bark extract using the cell growth, and also the skin in vitro skin. I will do the in vitro skin evaluations.
And you will not have major supportive data against analytical parameters that is required for your study. And what is this 940? You know, Carbopol 940. What do you mean by this 940?
940
Why
is
they
have
what is the difference? That's why I'm asking, what is the grade? What is the meaning of this 940? It's a grade.
This is a polymer grade, and this is a synthesized chemical, and that's why.
What it's mean? I know this is a grade.
Yes, sir.
Different grade and code will have some different meaning. And why you have not shown the flow property of your gel? It should be there.
Yes, sir. There is a flow property seen viscosity study for to measure the fluidity of my gel.
Okay, that is incentivized, correct?
Yes.
And out of these formulations, which one was having the best parameter as per you?
Yes, sir. I have also done the flux and permeability coefficient.
No, which one is best? You have 12 formulations, correct?
Okay, okay, sir. Well,
out of these 12 formulations, which one was the best?
Best one is, sir, F6, F7, F9, and F11.
Okay, and how we have performed the cumulative drug release study?
Sir, this is first time the in vitro permeability study to the skin layers. I have chosen the.
What instrument you have used?
A semi-polymer Milman, and after 1 hour gap, after 1 hour interval, the quantity of the polymer was taken within 1 ml solution. Then 1 ml solution was diluted. The fabricated.
Is there? Do you have used any transdiffusion cell, or you have used any semi-permeable membrane?
Sir, this is laboratory, this febrile diffusion cell consists of magnetic exterior, water bath, and the receptor compartment, the donor compartment. And the receptor
No. For cumulative study, just listen to me. For cumulative study, you have used semi-permeable membrane or you have used transdiffusion cell.
Sir, I have used
semi-permeable membrane, but you have not shown any activation data, because semi-permeable membrane, if you want to use, you have to activate it with some of the solvents like 1% EDTA or in water. Then the pore size will be open and you will get some permeation release or like cumulative drug release that you have not shown in your study.
Yes, sir, I have shown you, sir. This is the percentage permeability. I have first done the percentage permeability, and then I have shown you the flux and permeability coefficient study. And then I have gone for the percentage cumulative study.
And my last question, what is the rationality of your work?
Rationality of this work to make after developing this gel, to make this gel for socioeconomic value in a very low cost. This is the rationality of this work.
But Carbopol 940 itself is a higher cost chemical.
The Carbopol 940 gel, I have used here in a very dilute form, sir.
Okay, this is from my side.
Only the water solvent, and water is a very cheap solvent.
Okay, Shudipta, you have told regarding zero order release, but your therapeutic ingredient is an extract. So an extract will contain many constituents, right? So how can you say that your drug release is zero order or like this?
Ma'am, actually,
which ingredient you have measured in the extract?
Yes, I have only mentioned here the blank gel base, ma'am. I will do the next research work. I will do my development of gel. I will develop the gel with the neem bark extract. This is the pre-formulation step only, ma'am.
So you have mentioned regarding zero order release?
Yes, ma'am. I have mentioned here.
But you have not done it till now.
Ma'am, yes, ma'am. This is I have done only the polymer release. Okay. Polymer content release, ma'am.
Polymer release of zero order. Okay, you will be doing with neem bark extract. So which constituent you will do? How will you isolate that?
Neem bark extract, how I am isolate? Ma'am, I will prepare the extract, and then after preparing the extract, I will make the formulation. Antioxidant.
You are intending to use it as a topical gel over the skin?
Yes, ma'am, I will use this topical gel. It says, ma'am,
yes.
I want to also control release the
so you will be using it for topical gel. So actually the active ingredient should permeate. Active ingredient should get released from the gel, right? Yes. So where is that study? How you are measuring the entrance of the active ingredient from the gel onto the skin, into the skin? Where is that study?
In the study, we wanted to see that the research work for the polymer, it was used as a carrier to carry the extract. So we wanted to determine the polymer release contained into the solution. For this reason, I did this study, development of blank gel basis, and after that I will prepare the neem bark.
Polymer is not intended to give the effect. The drug is intended to give the effect.
Yes. Here in the subject of the study is only to predict the drug delivery system of this polymer gel. I didn't determine here any kinds of therapeutic effect because Carbopol 940 gel doesn't. I didn't see any kinds of biological activity regarding wood healing acid and everything.
I understand you have not done. That is why I am telling that what is the reason to present this, right? So okay. Thank you. Yeah, Ashim sir.
Yeah. Thank you, madam. Thank you, Ms. Sudipta. Can you stop your sharing? Please stop your sharing so that you can go ahead. So next will be Mr. Please stop sharing. Okay, fine. Thank you. So now the next presenter will be Mr. R. Rajagopalan. Mr. Gopalan, please start your presentation quickly and please follow the timeline.
Yes, sir.
So within 7 minutes, you have to complete your presentation. Start quickly. Please go ahead.
Sir, my PowerPoint is visible.
It is perfectly visible. You are audible. Please.
Yeah. Okay, sir. Thank you. So, good evening, everybody. I am R. Rajagopalan. I am here presenting the topic on surface modified 5-fluorouracil bearing liposomal gel anchored with this novel conjugate for treatment of actinic keratosis. So actinic keratosis, it is the precancerous of skin cancers, is developed the non-melanoma skin cancer. So it's a type of skin cancer. So commonly available drug in the actinic keratosis and the skin cancer is the 5-fluorouracil drugs is marketed available. The main problem is the 5-fluorouracil drug is here poor permeations in the skin. So marketed available drugs, it's not reaches the target sites at the good concentrations levels, and the maximum amount of drug it reaches the systemic circulations. So with regard to permeability improvements, we hypothesis such a study, such adjuvants, it is contain long hydrocarbon chains, desired hydrophobicity upon optimal absorption of the drug molecules. So we developed such a system, topical drug delivery in the field of actinic keratosis disease. Mainly it is produced in the lacuna in the synergistic effect in the marketed formulations. So in these studies, develop a synergistic cytotoxic activity on the lesion site, increase the localization of 5-fluorouracil drug at the tumor sites, and the same time reduce the systemic toxicity. For the purpose, develop the surface modified liposomal gels by the combination of using the gallic acid steer element conjugates. So here first time we synthesized this gallic acid steer element conjugate. The advantages of the synthesis method is we are using the direct amidation reagent at trifluoroethyl borates, thereby avoiding the tedious workup like the column chromatography and the aqueous workup procedures. So the synthesized conjugate is proved by the NMR studies and mass spectroscopy studies. So after synthesized conjugates, the conjugate is evaluated by cytotoxicity study using the A431 epidermal carcinoma skin cancer cell line. At the same time, the conjugate is studies by also the normal arcade cell line also. So the results are proved that the synthesized conjugates having the given IC30 value 10 microgram per ml, at the same time 5-fluorouracils was identified, the IC30 value was 1 microgram per ml, and also same times the in normal human arcade cells also the synthesized conjugates was proved that is less toxic, that is safe in the normal arcade cells also. So based on these studies, it showed that the prepared conjugates having the good specificity towards A4-3 cancer cells, and also same time it is safe in normal arcade cells. So it is the best choice of candidate for developing the topical drug delivery systems. After this, main advantage of the studies, combination therapy studies, the 5-fluorouracil and gallic acid steer element conjugates taken 9 different ratio in the ratio of 5:1 to 1:5. Performed cytotoxicity study, it was proved that the ratio of 1:1 was observed that give the synergistic cytotoxicity activity with compared to individual 5-fluorouracil drugs. It increased the results indicates that 2 folds increase the synergistic cytotoxic activity of 5-fluorouracils when combined with the prepared conjugate. At the same time, out of 9 ratio, over 9 ratio, the same studies was performed on the normal arcade cells also. It is proved that the ratio of 1:1 was given synergistic cytotoxic activity 60%. At the same times, it is less toxic in normal arcade cells also. So after the cytotoxicity studies, the formulations was developed. It was shown that the 4 different formulations was prepared in the various ratio of phospholipids and cholesterol ratio 9:1 to 6:4. It was shown that out of 4 formulations, 7:3 ratio is best, is showing the less vesicle size, good percentage encapsulation efficiency, zeta potential, and polydispersity index. After this liposomes is converted to gel, also showed that various characterization studies, spreadability, viscosity. So all are showing that the 7:3 ratio formulations was the best. So here the schematic representation is showing that our synthesized conjugate is attached with in the surface of the liposomes, because the gallic acid is projected towards the hydrophilic domain of the liposomes. The stereolamine is present in the between the interaction between the phospholipids of the molecules, and the 5-fluorouracil drugs is present in the aqueous core of the liposome system. So after that, it is proved on the ex vivo skin permeation studies, it is proved that the prepared formulations having the less flux compared to marketed available 5-fluorouracil formulations, it was shown that the marketed formulations having the more amount of drugs systemic absorbed and a very less amount of drugs is present in the target sites, that tumor sites. But our formulations, maximum amount of drugs presence in the tumor sites, very less amounts is reaches the systemic absorptions. So after that, the prepared formulations showing the cytotoxicity studies, the IC30 values was found that 0.5 microgram per ml. In the same, the individual 5-fluorouracil showing the 1 microgram per ml, the result shows that 50% increase the anti-cell proliferation activity of 5-fluorouracil liposomal gel. The next, the studies was proved that a skin localization index study using confocal laser scanning microscopy study, it is proved that the prepared formulations, maximum amount of fluorescence intensity was observed at viable epidermis part, because the actinic keratosis is the origin layer, is the basal epidermal layer of the skin. So it was proved that the maximum amount of fluorescence intensity is present on the viable epidermis, particularly the lower epidermis part of the skins. So the studies is proved that the maximum amount of skin localized delivery of 5-fluorouracil is present in the basal layer of the epidermal of the skin. So, and also it is proved that in vivo biodistribution studies, the gamma scintigraphy imaging and the distribution studies, the results was showing that the gamma scintigraphy images showing that the maximum amount of gamma, that radioactivity is present in the on-site of application only. So it is proved that the prepared formulations increase the site-specific delivery potential action at the tumor sites in compared to marketed formulations.
Last minute to complete your presentation.
Yes, sir. So the final conclusion of the work, it is showed that the prepared highly lipophilic, the conjugates, it's showing that prepared using direct amidation procedure, avoiding tedious workup. So we utilize the development of 5-fluorouracil liposomal gel, enhance the biocompatible potential. It is the safer approach treatment for actinic keratosis in comparison to marketed one. Yes, sir.
Is it over? Presentation done. Thank you. Thank you. Over to Saurabh sir, please go ahead with your query.
Yes, really good. And I'm impressed with your work actually. It's very good and all the data is there. Still, I have some questions. And in phospholipid, you have used only soy lecithin or you have tried with some other long chain fatty acid?
No, sir, I am tried only soy lecithin. I am tried.
You could have tried with like stearic acid or like palmitic acid. Then you could have come to the optimization part actually. And can you show me the release study slide actually? Whatever you have shown. Here, keep it.
Yeah, yes.
Here you can see that red one, correct? LSC formulations.
Yes, sir.
Which is having less cumulative drug release, correct?
Yeah, yes, sir.
And go to the previous slide. Here, in characterization part, you have marked that LS6, that third formulations,
yes, sir,
which is having less particle size.
Yeah, yes, sir.
Okay, I have doubt only in this part particularly. You are getting very good result here in these formulations, but comparison to the cumulative drug release, you are getting a very bad result with these formulations. How come this possible?
Sir,
here LSA is showing very good result in release study.
Yeah.
But you are not getting very good particle size compared to LSC. But it's quite good because these all are the below 200 nanometer.
Yeah, yes.
Do you have done any toxicity study?
Yes, sir. Yes, sir. Toxicity there is shown. Yes, sir.
You have not shown any histopathological study. This is the cell line study. What you have performed.
Sir. That work was done in Dabur Research Foundation. Here I am showing only that I am done is work in INMOS. I am done whatever only the result. Only.
And the cell line is available with them only, correct?
Yeah. Yes, sir. Yes.
Okay. Thank you from my side.
Okay, sir.
Madam, any comment on it?
Yeah, it is a very nice study. But can you just tell me how you have formulated the liposomal gel?
Yeah, I am formulated using that fast film iteration methods. In organic phase, they taken the soya lecithin and cholesterol and the prepared conjugate is mixed in the chloroform ethanol mixer. The aqueous phase is taken the 5-fluorouracil drugs with the phosphate buffer 7.4. The aqueous phase is transferred to the organic phase using rotary evaporator. After formation of thin film, the thin film formation after the aqueous, the pot is hydrated with the lipid layer. After this, it's going to probe sonigated. After probe sonigated, it reduces the vesicle size. After the formulations going to lyophilized the powder form.
Okay, so how stable is your formulation?
Yeah, I am done this stability studies also done as per ICH guidelines in 3 different temperatures that 25 degrees Celsius and 40 degrees Celsius and refrigerator conditions. It is showing that liposomal formulation showing the refrigerated conditions showing the good stability. But in gel formulation showing the 3 temperature, it was observed that good stability.
Yeah, good stability means what is the expected stability date, that is expiry date of your formulation?
So I was found that based on the results of the particle size and entrapment efficiency and spreadability, this type of data is not so much deviations coming from initial data. From this data, I am concluded that up to 6 months, my formulation is stable in the 3 different temperatures according to ICH guidelines.
Okay. Okay, thank you.
Okay, madam. Thank you.
Thank you, sir and madam. And thank you, Mr. Raja Gopalan. Kindly stop screen sharing option.
Yes, sir.
Next presenter will be Monalisha Mitra from BIT Mesra. So you can start your presentation quickly and follow the timeline given in the guidelines. Yeah, it's visible. Then make it full screen and start your presentation. Yeah, perfect.
Hello everyone, good afternoon. Myself Monalisha Mitra, currently pursuing my master's in Pharmaceutical Quality Assurance from Birla Institute of Technology. My topic for today's oral presentation is a review on carbon nanotubes and nanomedicine. So this is an overview of what I will be discussing today throughout my presentation. I will be covering these 4 parts in a detailed manner that are shown in these circles, and these are my references that have helped me to prepare this presentation. So the first circle gives us a basic introduction of what are carbon nanotubes, its structures, and its properties. So what are carbon nanotubes? They are basically long, thin cylinders of graphite, first discovered by Japanese scientist Ijima and its co-workers in 1991, and are now considered to be a top-class subject in academic researches as well as in various industrial areas. They are allotropes of carbon made of graphite and are constructed in cylindrical tubes with nanometer scale in diameter and several millimeters in length. The structure of carbon nanotubes are consisting of exclusively carbon atoms arranged in a series of condensed benzene rings rolled up in a tubular structure. They are hollow cylinders consisting of a hexagonal arrangement of sp2 hybridized carbon atoms and formed by rolling single or multiple layers of graphene sheets into similar cylinders. So CNTs are endowed with exceptionally high material properties such as electrical and thermal conductivity, strength, stiffness, toughness, and low density. The tensile strength of CNT is 100 times greater than that of steel, and the electrical and thermal conductivity approaches that of copper. This chart gives a detailed idea regarding the properties of carbon nanotubes. I'm not going into detailed discussion here because of time crunch. So the next part of my presentation deals with the classification of nanotubes. So carbon nanotubes are classified as per the following, this chart shown here. So the first is the single-walled carbon nanotubes, then is the multi-walled carbon nanotubes, after that double-walled carbon nanotubes, nanobuds, fullerene, and functionalized carbon nanotubes. So single-walled are basically one-dimensional. They consist of only one single layer. Multi-walled is two. It can be considered as a one small nanotube within a large one, or a roll of newspaper. And the nano double-walled is made of two. It is a hybrid of both the single-walled and multi-walled. Nanobuds is basically fullerene attached to a carbon nanotube, and functionalized ones are the activated ones. So my next part of presentation deals with the application of carbon nanotubes. Three applications that are dealt here are antimicrobial mechanism of carbon nanotubes, use in drug delivery, and as artificial implants. So antimicrobial mechanism of carbon nanotubes, reviewed literature shows that the size of carbon nanotubes plays an important role in the inactivation of the microorganisms. So various mechanisms that have been postulated till date are attachment of CNTs on microbial cell surface to promote the transmembrane electron transfer and induce cell wall membrane damage. Next is the protein dysfunction and DNA damage when CNTs penetrate the bacterial cell. And another is the formation of the secondary products such as reactive oxygen species. Some studies reported suggest that 60-minute contact time is enough for this destruction, whereas other studies are also there that suggest that longer duration of time, maybe up to days, is required to obtain the same result. And in case of the ROS generation or the reactive oxygen species generation, oxidative stress is considered as the main mechanism to induce toxicity of carbon nanotubes. When the carbon nanotubes penetrate the microbial cells, ROS as well as free radicals are generated. These free radicals then cause severe destruction of nucleic acids and lipoproteins. In carbon nanotubes and drug delivery, they are considered to be promising drug carriers and target drug delivery system. They can easily pass through the different biological barriers, pass through the plasma membrane, and enter the cytoplasm through tiny nanoneedle mechanism. They generally process using CNTs can be briefly summarized as drug is fixed on the surface or the inside of the functionalized carbon nanotubes. The conjugate obtained is then introduced into the animal body by classic ways or directly to the target site. For example, guided by an external magnet to the target organ. The cell ingests the drug CNT capsule, and finally the nanotube splits its contents into the cells, and thus the drug gets delivered. Then carbon nanotubes as artificial implants. The aim behind tissue engineering is the substitution of damaged or diseased tissue with biologic alternates that can ultimately repair normal and original function of the tissue. Due to post-administration pain, the body often shows rejection for implants, whereas because of miniature size, nanotubes easily get attached with other amino acids and can be used for implanting artificial joints without rejection reaction by the host. They can be incorporated in biomaterials supplied to bone mainly to improve their overall mechanical properties. They're expected to act as scaffold to guide and promote bone tissue generation. So my last part of today's presentation deals with health hazards related to carbon nanotubes. I will be focusing here mainly liver and renal system toxicity. So liver toxicity, liver is one of the significant organs in the body which plays a crucial role in carrying out various metabolic pathways in the body. Hence, any drug given in the body is processed through the liver either directly or indirectly. So it becomes imperative to study and analyze toxic effects of carbon nanotubes on liver tissue. Literature study reveals that accumulation of carbon nanotubes induce cell proliferation, inflammation, and increased amount of enzyme production. Histological study reveals the hepatotoxicity with the increased levels of bilirubinemia, which is an indication of liver damage. It shows damage.
Last minute to complete your presentation.
Yes, sir. It shows damaged Kupffer cells, which is an indication of liver damage. Next is the renal toxicity, as kidney plays an crucial role in excretion process. So toxicities should be studied. CNT accumulation leads to renal toxicity. CNT's own reaction with the cell shows increase in cellular apoptosis. Also, cell adhesive ability decreases. The cell cycle gets disrupted. So finally, I would conclude my today's presentation by mentioning the fact that science of nanomedicine has become an ever-growing field because of its unique properties. The graph shown here, obtained from the Polaris market research analysis, depicts the CNT market size and predicts its future, which clearly shows that it is surely going to increase in the upcoming days. But irrespective of the plethora of uses, their toxicity levels in human body have limits its wide-scale application. Thus, extensive research in reducing the toxicity levels must be carried out. Finally, thank you everyone for taking out the time to listen to me so patiently. Thank you.
Thank you very much, Monalisha. Over to Saroj Khatri.
So you have mentioned a lot of toxicity things actually, or risks in your presentations on carbon nanotubes. That means there are a lot of risk, correct?
Yes, sir.
So then why you have chosen this topic actually about carbon nanotubes? What is the rationality or novelty of your topic?
This graph shows here that it will be increasing. It has increased and it will be increasing. So
that is showing the market research actually, but not about the safety and efficacy.
No, sir, it is the USD million dollar, which it means by market size. So the toxicity is one factor that is limiting its widespread application. But if research on that domain is carried out, which can control its toxicity, it will surely be one of the boost or one of the highly demanding topic in pharmaceutical market.
And is they biodegradable?
Sir, I need to look into the literature. I'm not sure. I need to look into the literature.
We don't have any supportive data actually on biodegradable. It's compatible, but it's not biodegradable till then. And why it is called carbon nanotube?
So because of its it is in nanoscale range, means length is in nanometer.
Tell me the length, size.
So it ranges around 1 to 10 nanometer in size and several
1 to 10.
millimeter.
Sure.
So not sure.
Okay, this is from my side. Over to Momita.
Yes, Monalisa. What is the basic difference between a nanoparticle and a nanotube?
There's a basic difference between nanoparticle and nanotube.
Yes. Why you are calling it a nanotube?
Ma'am, this is because of its structure. It is tubular in shape. This nanotubes.
So what is the added advantage of having a nano formulation in the shape of a nanotube?
When the surface area increases, so the drug loading will be increasing. It can easily get into the targeted site.
Can you present any literature on that, whether the surface area is increasing or not?
Ma'am, in targets means when in case of drug delivery, we are seeing that carbon nanotubes is generally used for cancer treatments where targeted drug delivery is there. So it is a definite.
Okay. I think Ashim sir, we are running out of time. Yeah.
If panel permits, shall I ask one question?
Yes, sir.
Sure.
Yeah, Monalisha, you have mentioned in the presentation. You have mentioned in the presentation that these carbon nanotubes, they are acting as a carrier, and normally we are using this for targeted drug delivery. Now, my question is, how we will be incorporating the drug molecules into the carbon nanotubes, and what will be the size after drug incorporation or drug loading, so that it will be easily crossing through the various biological barriers?
So the last part that I have shown here, the functionalized carbon nanotubes. So the drug means it is the activated carbon nanotubes. It is get reacted. I don't know much about the size it will be after drug incorporation, but that's how we incorporate the drug.
Right. Okay.
And do you know any preparation method's name?
Yes, sir. Chemical vapor deposition method, laser arc method.
Okay. Thank you.
Thank you, sir.
Thank you, Mona Lisa, and thank you, sir and madam. So we will be continuing this session in another link, because in this same link there will be a plenary session. We'll be starting right from 3 o'clock. So we have overshoot the time little bit. Do not worry. There are 3 participants left. One is Dr. A. Gita Lakshmi ma'am, Deepan Roy, and Mr. Abhishek Jana. So we'll be providing a separate link, Zoom link, to them. You can join there within a couple of minutes. We'll be providing the link. The rest all can join the plenary session. Those who want to join the plenary session, there will be from 3 o'clock. So that will be going on in the same link. But the oral presentation for 3 candidates will be going on another separate link that will mail you shortly. Thank you. And now I would like to invite Pratibha madam. Pratibha madam, are you there? Can you hear me?
Yes, sir. Yes, sir, I'm
Okay.
here.
Okay, thank you, madam. Now the session, please carry on the session and go ahead with the plenary session. We are resuming from here. And participants, 3 participants, presenters, please join with us within a couple of minutes. Over to Pratibha madam. Thank you.
Thank you, sir. Thank you, sir. So with this, we have ended with our oral presentation. So now we are having our chairperson, Dr. Sanchita Ghosh, and our speaker for this session, Dr. Reema De. And very good afternoon. Very good afternoon once again to one and all present here. And thank you for joining us at the 2nd International Conference on Transforming Ripples in Healthcare Research, Obstacles, Sustenance, and Cutting-Edge Innovations. Myself, Pratibha Gupta, currently working as an assistant professor in School of Medical Sciences, Adams University. I would like to welcome our eminent speaker for the session, Dr. Reema De. She is a confident personality coach, image consultant from Jharkhand, India, and our chairperson, Dr. Sanchita Ghosh. She is from the Department of Psychology, West Bengal State University. It's my pleasure to introduce the session 2 of day 1 to you all. The title for a Lead Your Mind session is Emotional Agility, the Tool to Thrive in Uncertain Times. The mind is the primary domain of psychology. From rectifying problematic thought patterns to uncovering the workings of memory, mental illness, and emotions, psychology is heavily involved in the analysis of the mind. The mind has been a subject of debate for centuries. From now onwards, our chairperson for the session, Dr. Sanchita Ghosh, will walk you through the session. So I would like to hand over the stage to Dr. Sanchita Ghosh, ma'am, for conducting the session onwards. Over to you, ma'am.
Thank you, Pratibha ma'am. Am I audible?
Yes, ma'am, you are audible.
Okay. I'm really honored to be a part of SCRB DST sponsored second international conference on transforming ripples in healthcare research, obstacles, sustenance, and cutting edge innovations organized by School of Medical Sciences, Adamas University. Thank you, Dr. Partho Roy, convener of this conference, for this nice and interesting opportunity to be listening to a presentation on a topic that is gaining significance very fast. Gone are the days when people used to think that emotion is something restricted to just tearjerkers, be it in film, television, or in book, as the awareness of the individual.
Ma'am, you are not audible, ma'am.
Ma'am, I think, Sanchita ma'am, you're in mute.
Am I audible now?
Yeah, you're audible.
Yes, ma'am, now you are audible.
Okay. So from the very first, I was muted.
No.
Okay. So at first, I would like to express my gratitude to the convener, Dr. Partho Roy, for inviting me to chair the session. And this is the topic which is gaining significance very fast. Gone are the days when people used to think that emotion is something restricted to just tearjerkers, be it in film, television, or in a book. As the awareness of the individual and the collective psyche is on rise, the word emotion has appeared before us with a huge significance. Since the last few decades, EQ has been getting an important as IQ is in the social and professional field. We all know that things took up a sharp, in some senses, devastating turn after the planet has slipped into a grip of a deadly virus. Some words and the coinages like WFH or work from home, social distancing, lockdown have brought a huge change into our psychological fabric, both on the individual and the community level. With all the existing structures of work and work-related issues at different levels of life, going for a toss, emotional intelligence has come up as the tool to survive in such uncertain, unpredictable, and unnerving time. But what actually is this emotional intelligence? How does it enable someone to remain productive and cope with challenges cropping up from different corners? What kind of relations does it have with professional, fiercely competitive work setup? Our distinguished resource person for this session, Dr. Rima De, a renowned confident personality coach, would be throwing some invaluable light on such questions. A scientist by profession, Dr. Rima De has garnered a decade of rich experience in research and development. Her professional experience includes working as a scientist at General Electric Global Research, Scientist Fellow at CSIR National Metallurgical Laboratory. She is also an internationally certified image consultant. Through training or coaching sessions, she helps people to realize their true potential. Such personal discovery not only enlightens the individual, but also benefits the organization she or he is working in. Like all of you here, I am also eager to listen to Dr. De. Thanks, and please welcome Dr. Rima De. Over to you, ma'am.
Thank you, Sanchita ma'am, for that wonderful introduction. And thank you to Partho for having me for this session. Although I had that idea, I hope I'm audible, right? Just can you confirm me with the thumbs up?
Yeah,
I'm
it's audible.
Yes, yeah. Thank you so much. Thank you so much. So I had an idea. I somehow got an idea that this is all for students, but I see that there are some researchers and professors also attending this session. But I had planned activities for students, but I think it will be more or less relevant with for everybody. And with
No,
that,
just sorry to
yes.
interrupt, Rima,
Yeah.
that no worries, the topic you are speaking on, we are all students only.
Yes,
No
even
worries.
I myself is a student.
Yeah, fine,
Thank
fine.
you
Please carry
for
on.
giving that motivation. Yeah, I myself is a student. And of course, we have psychology professors over here. So I think they will have more information than me on that. But I will take you through this session in a way which is all my understandings that I have gained over the period of years through working in various domains and in various profiles. So with that, I'll start my presentation. Just let me know once my screen is visible. Is my screen visible? Yes?
Yes,
No?
ma'am. Yes, yes, it is
Okay,
perfectly
thank you.
visible.
Thank you. Okay, a little backstory over here. Back when I was in class 8, that was the first time I decided that I would participate in extempore. With so much courage, I participated in the extempore. And if any of you don't know what extempore is, just to quickly let you know, extempore is a session where you're given a topic just 5 minutes before you speak on that topic. So the topic that I was given was trees. It was such an easy topic. And as I walked into the classroom, a big, huge hall room actually, and I went there and said, "Good afternoon." And I said, "Trees are very important for us." And I just literally froze there. And I could see the students watching me and I watching them. And I could speak nothing. For 5 minutes, it was an absolute pause. And I walked out saying, "Thank you." So, what just happened? I couldn't face the fear of speaking publicly, known as glossophobia. It's one of the highest rated fears, and it just took over me. Fear was an emotion there which just took over me so much so that it was so paralyzing that I couldn't even speak on a topic like trees. And that applies to our workspace as a student and as a professional. We think that we are logically driven, but we forget that we are biologically woven. And that's the reason we are here to discuss the topic emotional agility, the tool to thrive in uncertain times. So the ground rules for this session is be respectful, be interactive. I want you all to be respectful so that we are not judgmental. For some moments, we are very judgmental beings. So don't be judgmental in this session. Let it be a safe space for everybody to open their minds out. And please participate in the polls and activities that would make the session more interesting. And I have some activities written down here so that I don't forget doing it. That's the reason I'll be referring to my notes in between. So what's the emotional temperature of the room right now before I move ahead? Is everybody relaxed? Is everybody happy? Is anybody there over here who is tensed? I'll just stop sharing my screen. I'll come back here. I would request all of you to be on camera. I don't care what your background is, but I want everybody over here to connect. And if possible, students and the professors, I'll request everybody to be on camera. Thank you so much, Joyita. Thank you so much, Shrija. Thank you, Priyanka. Thank you. Thank you. So what's the emotional temperature here? Please unmute yourself and say, are you feeling relaxed? Are you feeling tensed? How is it?
Relaxed.
Quickly, if I can have some inputs.
Yeah, relaxed.
Relaxed, great. Joyita, great. Thank you for that input. Anybody?
I would say I was tensed before this session, but now I'm relaxed.
Okay, you are anchoring for the first time, is it, Pratibha? Is it? Okay, okay.
No,
And you're doing great. Let me tell you that. Okay.
Thank you. Thank you.
Okay. Okay, so let's just go back to the presentation. Okay, is my screen visible again? Yeah?
Yes.
A quick yes? Okay, thank you so much. Why isn't it moving? Yeah. So why do we need emotional intelligence? I have designed it from a student context, but it applies to all of us. So let's try to understand how is emotional agility important. For that, let's first try to understand one thing. Imagine that you are taking steps and you're each 1 meter, right? So, which is the length of your each stride is just 1 meter. So by the end of 30th meter, by the end of 30th step, I'm sorry, where would you reach? By the end of 30th step, where would you reach? Quickly. Is that such a difficult question? There are more difficult questions to come if you're finding this difficult. Okay, by the end of 30 steps, we would reach 30 meters. All of you agree on that? Give me a quick yes in the chat if yes. I would be happy to get that yes. Yes? Okay, I can see only Sanchita, Pratibha and Partho on my screen. So I can get it from their head nods. Okay, let me see, there's something in the chat. Okay, thank you, Shrija, and thank you, Vitasta. Thank you, Dr. Anshu Ali. Okay, now what happens if I say that your stride doubles every time you take a step? That means that first step you take is 1 meter, the second step you take is 2 meters, the third step you take is 4 meters, and the fourth step you take is 8 meters. So where will you be by the end of 30th step? Difficult to calculate, right? Right? Now, to give you an answer to that, that is equivalent to 26 times of rotations of the Earth. By the end of 30th step, if you're exponentially taking steps, you would reach, you can take 26 rotations of the Earth. Now, how is it relevant here? Well, if you are a student, all this while, you were doing studies, class 1 to 12, you were doing your BTech or BSc or MSc degree, you knew the end result. It was a very linear thing. You could calculate in your mind that by the end of this, this is where I'm going to reach, right? But when you step out of that zone, it's no more linear. It's actually exponential. You are stepping out from a place which has linear results into a place which is kind of exponential. So the work dynamics that you're stepping into, your accountability goes up way higher, right, than you were a student. And that is the reason you are in a space, you don't know how to handle everything around you when you are starting. Even if you're not starting, the technological advances that we have made in the last 100 years, say 1900 to 2000, that is something we are making in just 4 years now. And this is a prediction by Peter Diamandis, the futurist, the well-renowned futurist, that this technology evolution has taken up an exponential curve and it is going to affect us all. And when we are growing exponentially, we don't know where we are going to land. And when we don't know where we are going to land, we don't know what the situation we will be in. So how does it, again, I'll give a metaphor here. How does it show up? In that case, it's something like that, that for students, it's a transitioning happening from my ecosystem where they were in a pond, right? If I can say that your college was a pond or a lake that you were thriving in, you were doing very good, but all of a sudden, you have to go into a work scenario, and that is like a huge ocean that you have been poured into. So your swimming skills that you were using in the pond no more apply there, or rather they apply, but you have to upskill on that. Just with that swimming skill, you may not survive in that sea, right? So for that, there are many things you might have to work on, but one of the most important things that you can work on is your emotional intelligence. Now, before I speak about emotional intelligence, what is the world saying about emotional intelligence? Now, these are some statistics collected from various credible sources, and it says that 90% of top performers have high EQ. Indeed, any leader who is very well renowned, irrespective of whether she's a male or she's a female or he's a male, they are having equivalent EQ. Okay. Now, EQ is also responsible for 58% of your job response. And in US, the statistics shows that when you have a higher EQ, you are earning 29,000 more in terms of dollars annually. Also, these are some surveys done by different companies, EQ at Work, and one done by Berkeley UC says that emotional intelligence is 4 times more powerful in predicting your success. Then a study done on managers within PepsiCo shows that managers with high IQ perform better. And then again, Motorola has done a study where they see that the productivity goes up post an emotional intelligence program. And World Economic Forum had predicted long back that by 2021, EI will be in one of the top skills for job, and which has already happened. I don't need to say that. With the COVID happening, we all know what happened, and we all needed to gear up on our EI skills. So what does EI help in? It helps in our endurance capability. All of you agree? What is endurance capability? It is our ability to hold on and be persistent, be consistent, keep learning, keep improving, and keep growing. Now, in this equation, there is EQ and IQ. So can anyone quickly help me? What is EQ and IQ? Can anyone quickly help me? If you are interactive, it gives me the energy to carry on. And if you are not, still I would carry on, but it will be really helpful.
Ma'am, EQ is emotional quotient, I think.
Very right.
And
is that
IQ...
Bitosta?
Yeah, ma'am. Yeah.
Yeah, sure, Bitosta. Go ahead.
And IQ means intelligence quotient.
Absolutely right. So EQ is just a parallel of IQ, just to equate it. Now, right now, here we will refer it to as EI. So what EI exactly is? EI is our ability to recognize, reason, comprehend, and effectively manage emotions. And we will be doing relevant activities to understand it from a student context. And you can understand it from a professional context as well. I thought there are more of students here, so I had activities planned around them. However, this applies to everybody. Okay. So before we move any further, now I want you to do me a favor. All of you ready? Can you see my screen? Yes? Yes. Okay. I want you to do me a favor. I want to name these objects, and you will have to help me to name these objects. Will you do me that favor? Yes? No? Yes? You don't want to? Then let me move ahead. Okay. So then, assuming that as a yes, what I want you to do is, I want you to name these objects. I'll give you the name options. And once I give you the name options, whichever is the first one that comes to your mind, within the first 2 to 3 seconds of me giving the answer, me giving the name options, whichever is the first one that comes to your mind is the right one. Let me tell you that. Okay. Are the instructions clear enough? Can anyone just confirm? Yes? Is the instruction clear?
Yes, ma'am.
Okay. Okay. Thank you. Thank you, Joyita. So what I want you to do is, I want you to name them, and the name options are Kiki and Buba. So which is the one are you going to name Kiki, and which is the one you're going to name Buba? Whatever comes to your mind in the first 2 seconds is the right answer.
And that sharp one is Tiki and that one is Buba.
Okay. Okay. Now tell me, what do you think? Who was that, by the way? Can I just know your name? I can't see you in screen. I'm sorry.
Ma'am, I am Irfanullah, B Farm 4th year.
Okay. Okay. Irfanullah, right? Am I spelling it right, Irfanullah?
Yes, ma'am.
Okay. Thank you, Irfanullah. Now tell me, how many of you think over here that everybody would agree with Irfanullah? Matlab, everybody would name the first object as Kiki, the star object as Kiki. How many think it will be 100% people will name the first object as Kiki? Quickly, can you give me a number here? That no, 50% will name it as Kiki, 60% will name it as Kiki, 70% will name it as Kiki. What? In the chat box, you can give me a number if you don't want to speak up. Let me have a look on the chat box. Agree. Okay, Arijit says agree. Give me a number. What do you think? How many people in this? We have some 62 people. So how many of them would agree on this? 80% would agree. Okay, Jaita says 80%. Anybody else wants to give a number here? No? Okay, fine. So let me tell you, the number would be 98%. This is across the world. 98% of the people will name it as Kiki. Why? Because our cognitive abilities are the same. We process this information, we associate this intense, sharp object with K, which is K, which is intense. That's how we all have been brought up. So 98% will answer it as Kiki, but the rest 2% may be people who are specially abled and they do not process information this way, which brings us to the point that psychologically, our psychological makeup consists of our rational brain, our emotional brain, and then our personality. Now, more and more of research shows that, and Dr. Sanchita may be able to confirm on that. This is my study, that our intelligence quotient remains same over a period of time. It doesn't increase. But it's our EQ which we can work on and we can increase to become better professionals and better humans, leading life in a much more meaningful manner and in an impactful manner over a period of time. Now, again, this is by Daniel Goleman. He mentions in his book, although IQ is constant, but if you work on your EQ, there is a possibility that you can increase your IQ as well. Okay, so of all these three, personality comes from your genes, like 50 to 55%, but it's your EQ that you can increase very much to perform in the way that you want to see yourself perform. Okay, so what exactly are aspects of EQ? Now, there are 5 aspects of EQ, and I just took 4 over here again, considering that this is mainly a student session. So there are 4 aspects: self-awareness, self-motivation, relationship management, and empathy. And each of these aspects we're going to understand again by doing a couple of activities. So let's start with self-awareness. So anyone wants to say anything about self-awareness over here? Quickly. Anybody? What does the picture say? What comes to your mind when you think of self-awareness?
Ma'am, when you are aware about yourself, your inner nature, or like how you are as a person.
Okay. So self-awareness has many different meanings for different people, but in the emotional context, what is it? Okay. So one of my favorite quotes about self-awareness is, being self-aware is not the absence of mistakes, but the ability to learn and correct them. Now, what does that mean? Again, we have an activity here. I want you to say silk with me for 5 times. Now you all have to unmute yourself. I'm sorry. So I'll just stop sharing the screen here. And I would request you all to unmute yourself and say silk 5 times with me. If on my orders, if I may say so, I'm sorry, I don't have a better word right now. So on my orders, please say silk with me 5 times. Please unmute yourself. I'll request all of you humbly to unmute yourself. So may I start?
Yes, ma'am.
Okay. Silk.
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 Silk. Silk. What does cow drink?
Milk.
Grass. Grass.
Cow drinks grass? That is a new one for me.
Grass or water, not milk.
Right. So congratulations to all of you who said milk. That is the wrong answer. Of course, you know it. But congratulations to all of you who said water. Okay. Now, what happened here? Why did you say milk? And why didn't you say water? Perhaps if I had asked you the question, what does cow drink, without telling the silk, a little bit of silk backstory, not giving that context, maybe you would have answered water, right? The possibility of you answering water would have been much higher, right? But what happened here? Let me again just share my screen. So Has my slide changed? Yes?
Yes, ma'am.
Okay, thank you, Pratibha, so much. So what happened here? Now, according to Daniel Kahneman, he is also a well-renowned psychologist and an economist. There are two ways we think. We think slow, we think as fast as well. And both are needed. You need to solve your research problem. You need to be slow on thinking. You need to make your presentation. You need to think deliberately and logically, right? But at the same time, if you need to cross the road and you see a car approaching and you start there deciding it's going to come at the rate of this and it's going to hit me at this impetus or force, then you're definitely going to be get hit by the car, right? You want to take decision fast there. So we need the both ways of thinking. But now what happened is I prepped you up that silk. I gave you that information. And when I asked you the question, what does cow drink? Automatically, you had an intuitive reaction that it's milk. So you reacted there. Had you stopped for a moment and not reacted, rather responded, I stress on the word responded, you would have answered water. All of you agree on that? Yes or no on the chat box, please? Yes, thank you so much. So self-awareness is about knowing when are we responding and when are we actually reacting. Okay, now again, to give a little bit more understanding here, I have it in terms of a question format that I have with you. All of you have your smartphones with you, right? Yes. So if you just hold the smartphone on this QR code, you will see a question pop up on your screen. You can just rate yourself based on what you feel most. Rate these statements on a scale of 1 to 10 on what is true for you. Okay, so I'll show you the questions. And in case if you have, have all of you seen the QR code? It's opening for you. Can someone confirm me? Is the QR code opening?
Yes, ma'am, it's opening.
Yes, ma'am.
Okay. Then please put in your votes and let me share my screen again in that case. Let me just have a look whether I received the votes on that. So I haven't received any vote yet. So I'll share the link here again. Okay. I've received a couple of them. And again, I'll share the link here in the chat box. If any of you are not able to open the QR code, you can share it through here. You can vote through the link on the chat box as well. And let me share it with you, the vote results. Let me again start sharing my screen. Can you see? Can you see the vote results? I feel distracted quite often. That's true for, both is true for most of you. This gives an average result. So it's an average result of whatever. 6 of you voted. Come on, the number can be bigger, right? There are 66 people out here, right? I expect the number to be a little bit better. If you could do better, that would be great. So I have shared the link in the chat box and also the QR code. You can go onto this through any of it. And what this shows basically is an average score. If someone has a higher score, it will average it all out. So I feel distracted quite often and I feel irritated quite often. Why this happens and how can we reduce this? Again, let me go back to the presentation. Okay, so again, why self-awareness kicks in over here? Why do you feel distracted? Now, how do you feel distracted? It's either you start eating something, your brain wants to eat something. it asks you to eat something, or you go on your phone and you scroll through the feed or anything. You go and chat with a friend, or when you're supposed to work, you go and chat with a friend, right? Why do you feel distracted? Just before the distraction happening with you, if you have observed yourself, you will realize that you are either feeling overwhelmed by what is happening around you. It may be something like this, that the COVID scenario, you don't know how your job will be and you're feeling overwhelmed. And that's the reason you want to just numb that feeling down and go on something else. You go on social media and you forget that feeling. You want to make yourself feel better. And that is also the reason that we are binge eating many a times. We are eating more than what is required, because that's a way our brain reward centers get activated. So when you know what is the feeling behind you getting distracted, you know how to address that feeling right at its root. And when you feel distracted, you also start procrastinating. And when you start procrastinating, you also start feeling guilty. When you start feeling guilty, you also start feeling irritated. And when you start feeling irritated, you don't take that irritation out in a meaningful manner, but maybe you take it out on your nearest family members, because they are the people in our life we take for granted, right? So if you can address the problem right at the root, why you're feeling irritated, why you're feeling distracted, then you know how to address the problem. And that is the reason self-awareness is so important. Okay, so let's move to the second one, which is self-motivation. Now this applies to all of us, right? Life will have its own curveballs being thrown at us. Sometimes things will go according to us. Sometimes they might not go according to us. But how do we keep ourselves motivated? How do we pick ourselves from issues that are very difficult? From a student context, it may be like something you didn't score very well, or you couldn't get the job that you wanted to. And from a professional perspective, maybe you were looking forward to that promotion, you didn't get that promotion, or maybe you were looking forward to that recognition that didn't happen. And this all somewhere hurts you. It makes you feel not happy about yourself. So how do you keep yourself motivated in those times? That's an important aspect of AI. Now that can be a whole chapter in itself, but I will just explain one of the ways that you can self-motivate yourself is by being not self-critical, but by being self-compassionate. Now, all of you must have heard what self-compassionate being is. If you haven't heard, then self-compassionate is about being kind to your own self. Now, what does being kind to your own self mean? Say you had a presentation coming up and you messed up. Do you say yourself, oh, it's okay, it happens, take it casually, it happens? That's not being kind to yourself. That's not what we mean by being self-compassionate. You say yourself something like this. Say if my session goes bad today and I say, Rima, I know you did certain things wrong. Let's sit down and write what are those certain things that went wrong so that we can take care of it in the next session. So this is something called self-compassion, where I am speaking to myself in third person. Why in third person? Why not I have done this wrong? I'm not talking like that. Why am I speaking to myself in third person? Because again, research has extensively shown that when you are being self-compassionate and you speak to yourself in third person, you are able to disassociate yourself from the person you are being. You can see yourself from a distance and you can rectify your problems in a better manner. So that's one of the ways you can work to motivate yourself when things are not right. With that, let's move to the other aspect of EI, which is relationship management. Now, any of you as a student, as a professional, tell me what are the relationships that you're managing in your workspace and in your student space. I'll again stop sharing here and I would like to hear from you all. What are the relationships you're managing? Are there any students here? I see everybody doctor, and
So you can unmute yourself and you can answer the question.
I'd love to hear from the students. Am I too scary? The students, I want to ask you, am I too scary? No, right? I also think so. So you can give me your answers. Okay, please quickly tell me, what do you think is the relationships you're handling in your workspace?
You can speak about your teachers also, though
Yes.
your teachers are present here.
Right.
But if you are facing any kind of difficulty, or if you have any relationship issue, you can share it with our resource person.
Thank you so much, Sanchita, for doing that. Okay, I see students are not willing to speak. So I think then you have no relationship issues, relationship management issues. Let's end the session here, right? No one here is having any emotional intelligence requirement, I see, okay? But still, I'll continue. So what happens is, when it comes to, let me just share my screen again. Okay, so when it comes to relationship management, now I had put up this question in the context of students. We go to the next slide. Yeah, what are the various relationships you manage as a student? I had put up this question in the context of students, but again, what I would like to say is, as a professional also, you will have to have different relationships. There are relationships which are above you. Maybe it's your boss you have to report to. There are relationships below you. Maybe it's students that you're handling, and there are relationships around you. It's in horizontal relationships like your peers, your colleagues, and everybody. Now, for students, you have relationships to maintain with your classmates, which is an horizontal connection. And then you have relationship to maintain with your juniors. And if you are a junior, then no juniors under you. Sorry about that, can't do anything about it. But you will have your seniors that you'll have to have relationships maintained with, and you will have your professors that you'll have to communicate with. Now, you have a whole range of people that you have to deal with, right? Now, some of them are people that you get very easily along. Some of them are people you are scared of because they somehow intimidate you, they scare you, right? And some of them are people that you get along easily, right? Now, you would want to stick on to the person that you want to get along with easily because he creates or she creates a comfort zone for you, right? Now, that you can easily do as a student in a college space. But ask your professors, as you move on to a professional space, you have to deal around with every kind of people because your accountability goes up. You cannot go and complain to anybody. This person behaves like this. That is the reason I'll not work with that person. This person doesn't know how to talk. That is the reason I will not work with this person. You can't do that. You have to be a team player. You have to understand what is the people dynamics around you, and you have to mold yourself accordingly in order to rise above in the leadership ladder. Every leader who is become a leader is a people person. He knows how to manage people around them. So that's one of the skills that EI again teaches you. Now, coming to the last aspect, which is empathy. Now, again, I would love to hear from all of you. When I say empathy, what do you understand by empathy? I'd love to hear.
Understanding and respecting the pain the other person is feeling.
Okay. But how does it apply to workplace?
It's like understanding the emotional or the emotions that the student is going through, or my peer is going through, or the state of mind my boss maybe is going through.
Right. Thank you, Joita, so much for that input. That's absolutely right. input, and I'll just give some examples how it applies to workspace. Now, empathy is something is like a 4-year-old kid. For him or for her, his toy breaking is a big problem. You know, it's a very big problem because his entire world is about that problem. Now, me as a mother, if I look at it, like, oh, don't worry, this is a small thing. I can't say that to him, right? I have to look at the problem through his lens or her lens. If I can't look at the problem through his or her lens, I may not be able to connect with that person. I'm pushing that person away, right? You get judgmental many a times, right? This is easy. Why you can't do it, right? Now that person's world is entirely different from ours. What is important for us may not be important for them, right? That is where empathy comes in. Now, again, I thought this is for students, so I had put up a mail, and I need a volunteer here now, a student volunteer. Please, someone who is going to read the mails for me. Anybody? I'm not going to eat you.
So
yes, ma'am. This is Amartya.
Amartya? Okay, Amartya. Amartya, I want you to do me a favor. Okay, I want you to read the email that I have put up in the next slide. And then there's also a following email. I want you to read both of them. Okay, Amartya?
Okay, ma'am.
Okay, so Amartya, let's assume it's you who is writing the mail. I know you're not this bad, but let's assume for a moment, okay? So read the mail for me, Amartya.
Hi, Professor ABC. Subject, urgently want to meet you. I want to meet you in person and discuss my issues. Let me know when we can do so. Regards, XYZ.
Now, is there anything wrong with Amartya's mail? Can anyone tell me? Is there anything wrong?
Ma'am, the subject is not correct. Urgently want to meet you. That it is wrong.
Who is that? I can't see you on screen. My apology. Vitasta. Okay. Why is the subject not correct?
Ma'am, because it is appearing as if he is directly communicating to his teacher. Like urgently want to meet you should be in the body of the mail, not in the subject.
Okay, fine. Is there anything else wrong? Thank you, Bitasta, so much for that input. Is there anything else wrong?
Ma'am, it is more sound.
I don't think there is anything wrong in this. I couldn't find anything wrong in this mail.
I find it that it is kind of order he is giving.
Yeah, who is that? Amartya, is it?
No, ma'am, it is Suraj Shah, ma'am.
Okay, Suraj Shah. Okay, yes, it kind of is the order. Now let's understand the difference, okay? Amartya, are you ready?
Yes, ma'am.
I'm sorry for making you look so bad, but I'm going to make you look good now. You're going to write a very good mail now. So the next, I'm going to change the slide. So please read out the mail. Okay, Amartya, go ahead. Now read.
Dear Professor ABC, subject is request for a meeting. I wanted to meet you in person to discuss some aspects of my assignments that I could not resolve. Being aware of your time schedules, wanted to mention that the problem would take only 15 minutes of your time. So I kindly request you to let me know a convenient time at your end so that I can schedule a meeting. Regards, XYZ.
So Amartya has become a good student now, na? What changed?
Yes, more polite.
More polite. What changed? What changed? What else? Anybody wants to add? What else changed?
The topic. Yes, I think the topic is much more discussed. Like
he has given.
This is Srija here.
Okay, Srija, go ahead. Yeah.
Yes, actually, he has given the detailed explanation of why he wants to meet, so that the professor or whosoever he is contacting also understands the importance.
Exactly. So there are many aspects. Now I'll tell you how empathy is working here. Now Amartya, when he is writing the mail, he is understanding that the professor is having a very tight time schedule. So first he mentions that it will take only 15 minutes of your time, right? So the professor can accordingly manage the time. Second, he also mentions that what is it regarding? It's regarding his assignment, which he did not mention in the previous mail. That makes the professor make up his mind. Otherwise, say, imagine I get a mail from somebody urgent. That may be urgent for him, but may not be for me, right? That may put me also in the panic mode. Like, what is so urgent, right? And as Shrija said, if I remember it right, it was more of an order kind of a thing, or I don't know who said it, my apologies. So the previous mail was
Suraj.
more of an Suraj. Oh, Suraj. Okay, I see you on screen, Suraj. Thank you. The previous mail was more of an order, but this is more of a polite thing. Now, understand one thing. Why do we need mails in empathy in emails? Now, email is a writing form of expression, right? The person on the other end is not able to hear you, is not able to see you. The person is only reading your words, right? And the words the person reads gives a tone, like what kind of a person you are, right? The words you read through, you understand the tone of writing is this. The previous mail sounded a bit rude, right? It sounded a bit casual, right? But this one is sounding much more polite. It gives space to the person on the other hand to understand what the situation is. How is it relevant in workspace? Again, in workspace, be it an academic scenario, be it a professional space, you will have to deal with so many clients, customers, people outside of your domain. They might not be knowing you. They communicate only through emails. And when there's an email communication happening, it's very important that you keep the tone very neutral, if not polite, but neutral. It should never go, rude until unless absolutely required. There are certain instances where it is supposed to be rude, but then you have to keep it polite because you have to work with people around. So was that activity on empathy useful? Yes. Did that give an idea how it's useful in the workspace? Yes, no, a quick yes, no would be really appreciated. Yeah. Okay. So with that, we have almost covered all of this. So now tell me, you are good in self-awareness, you are great in self-motivation, you're great in relationship management, you're great in empathy. Now, if you cover all of these 4 aspects in you, does that make you a better student? Does that make you a better communicator? Does that make you a better future professional? Does it? Yes, no?
Yes, ma'am.
Yes, okay. Can I hear from the professors as well? I'm sorry for asking that, but can I hear from the professors as well if you're around? Yes, no?
Yes, ma'am.
Yes,
okay. Yes. Thank you so much. Thank you so much. So now, what is it that is working in this case? What is it? I spoke about 2 words in the beginning, right? Emotional dash, the tool to thrive in dash times. Can someone help me? What was it? Emotional dash. What was the word?
Emotional quotient you were telling.
Emotional agility, right? And the tool to thrive in dash times. What was it? Uncertain times, right? Now, why did I speak about agility? It's because all of this makes you agile. What does agile mean? Agile means that you are flexible. You can take care of the situation, whatever it be, you can adapt to the situation, right? Now, again, from a student context, if we put all of this together, what happens in an interview scenario? Or if you're a professional already, you can consider this to be you presenting in front of other colleagues or staffs. What happens in an interview, although I've put it, you can extend it to presentation skills, that you have gone out there and you want to present something as an interviewee or as a professional, whatever be the case, but somehow we have that blood gushing through our hearts, our heart pounding before presentations, right? All of you had that feeling before presentations? You could hear your heart pounding very hard. Let me tell you, That's also called this syndrome called Takotsubo syndrome. Okay, the heart actually changes shape during events like this. Every public speaking event that you have to do, the heart might change shape because it's preparing you for something which requires a lot of your focus, and you would be the center of attention for people, and that puts you in a place where you don't want to be, right? But then you have to carry it forward. So you can't let that emotion to take over you. Right before the presentation, that heart pounding that you have to ensure, oh my God, this is happening. This is prepping me up for something good. I have to be super focused. I have to deliver. But instead, we tend to think, oh my God, I'm feeling nervous. I don't know, I will be able to do it or not. That's what we think, but that is not what we are supposed to do, because once we walk into the room, it shows up in our body language. Our mind and our body are connected. Even though you think that I will do it, but you go there and you are somehow like, if right now, if I am presenting somehow like this, yes, I know I am speaking right, but I don't know whether it will work, but I know this works. Is my body language supporting what I'm saying? Would you believe me? You won't, right? Even though I'm saying the right thing, because I'm not having that conviction, I'm not having that congruency. So EI applies everywhere. If you know how to take care of your nervous knockdown that happens before a presentation or an interview, you know how to put forth your point in a very presentable and confident manner. Okay. Now with that, I would request you all again to put up a word of how was your experience of this session. And I will take question and answers over here if you have any. And I would love to take question and answers. And after that, I'll share a resource which may be useful for all of you. So again, have you seen are you able to access the QR code? Yes, no? Let me just change the question here. Just give me a second. Are you able to access? Can someone confirm me?
Oh, ma'am, can you just share the QR code once again?
Yes, I'll share the link as well. I'll share the QR code as well. Where is the chat window gone? Here's the link, and let me just share my screen one more time. Can you see this QR code now?
Yes, ma'am.
Okay.
It is visible.
Okay, fine. So please let me know what was your experience of this session. In the meantime, please let me know if you have any questions from this session. No questions?
If anyone is having any question, you can unmute yourself and ask your question.
Good afternoon, ma'am. This is Amartya again.
Yeah, Amartya,
I want to ask you a question. Like, I have a very serious problem, which is the fear of being judged by the people. So, how to get rid of this? If you can help me out.
Amartya, that's not your problem alone. That's my problem as well. The fear of being judged by people is a universal problem. You can't get rid of it in a day, but what you have to do over a period of time is you have to understand that why do we feel this fear of being judged by other people? Because at the end of the day, all of us want to feel accepted. We want to be known. We want to be recognized for what we have done. We don't want to go wrong. We don't want to take up that judgment, you're not good enough. You're not doing that well. Our brains are already negatively biased. And when we hear anything like that, it becomes very difficult for us to use that as a resource and use it for our own good. Now, here again, the EI comes in. If you treat that fear of judgment by people as a feedback, that people are giving you feedback, that this is what is not going right, this is what is not right, you can detach yourself from that fear, not entirely, but to an extent, and you can use it as a feedback to get better and better and better. And this is also to do with your image. What do you think of yourself? Do you think of yourself as someone who needs constant validation? Like every time there is a validation coming from outside, you are going to feel good. That's not going to happen always. So you have to understand that you have to rely on your own validation as well. How do you do that? You allow people to judge you. You keep taking that feedback and use it for your advantage. It will not happen in a day, but if you open up that space and keep doing this, it will happen over a period of time where you will understand that you will fear the judgment of people, but anyways, you will do what you want to do. Was that helpful, Amartya?
Yes, ma'am, of course. Thank you so much, ma'am.
Okay.
One thing I want to add, ma'am, here.
I just want to, Srija, if I may interfere there.
Yes.
I just want to share the screen with all of you. Thank you for being so kind to me. Can you see my screen? Yes. Thank you for those inputs again. So these are 6 people only. Last time also it was 6 people. These are some 6 people who found me interesting. I guess the rest, I don't know what happened. And but thank you for this amazing input. And yes, Srija, you can now go ahead with your question.
Yes, ma'am. Thank you
also,
for
Srija, just a minute. I'll just share a resource here again in the chat box. Give me a minute. So Amartya, for you, this is a feedback form, a Google feedback form. I collect feedback from all of my every sessions to understand how it went, what went wrong, what went right. So you can give me a feedback there, and that would be immensely helpful for me to improve my work. And Amartya, see, I might get some feedback which may not be great, but still I have to take it. Right, Amartya, are you there?
Yes, ma'am.
Thank you, Amartya, for that wonderful question and being so brave of you today to ask that question. Yes, Srija, go ahead.
Thank you so much for the, first of all, thank you so much for the wonderful session, ma'am. Actually, these are the topics which I'm very interested about, and I really want to work towards these kind of, mental health, emotional health related issues. So recently, I have been associated with the Global Shapers Dusseldorf. So it is one of my friends' initiative. She started a pro bono initiative regarding mental health. The initiative is named as Vasudhaiva Kutumbakam. So in that we cater all these kind of mental health, and there are mental health issues, and there are a lot of people who are, since we are not directly from the psychology background, so there are people who are working in the psychology background, and they are technical people who can deal with mental health issues. So attending your session, it was really good for me. So I would talk with my friend, and I would really we would be really happy to collaborate, and we'll get to you regarding that. So if you can contribute in one of our sessions in Vasudhaiva Kutumbakam.
Absolutely, I'd love to, Srija. Thank you for giving me that opportunity. You thought that I'm worthy of it. Thank you for that, Srija.
Ma'am, I just wanted to ask one question.
Yes, Ditasta, go ahead, please.
Yeah, ma'am. Actually, ma'am, I face this problem very often. So related to that only, I want to ask that many a times this happens that there are two important, very, very important things in your life, and you are stuck between those two things. You have to select any one out of that. So I am actually not able to take the decision on the spot. I'm totally get confused and I just stop thinking. I just think, okay, go with the flow. I just get very anxious. So how to deal with such types of things?
You know, the number one reason we get anxious when there are decisions to be made, each of them may be equally good, and you don't want to let go any of them, because any decision that you would take will have its consequences. Now you are attaching yourself to the consequences. You're thinking about the consequences. If I don't take this, this would happen, this would happen. So if you're stuck on the consequences, it's again a negativity bias that's happening. You know, you're stuck there. It will be difficult for you to get rid of, it'll be difficult for you to make that decision. Work on what is good in both the decisions. Write down the points. What are the good things that are going to happen if you take decision 1 and decision 2? What are the good things? I'm very sure you will be able to figure out the good things that will happen, and then weigh them, you know, which is the better thing that is happening. I'm sure you'll come out with something.
Long run, with respect to long run, which will be
absolutely. Thank you so much for adding that, you know, long run. You know, right now we are staying in an immediate, instant result environment, and anything that is instant and immediate is never helpful in the long run. If you keep eating Maggi noodles every day, it's not going to help you, right? Same, always think in the long run. Delayed result environment, you know, always nurture that, because that is going to pay you in the long run. Because we see so much happening around us every day. Somebody is doing something very great and you think, where am I? What am I doing? I'm doing nothing. So that is one thing that takes over all of us. Stop doing that. Think in long term. Stay in a delayed result environment. You will flourish in the long term. Always long term. Thank you so much, Vitasta, for bringing that point up.
Thank you so much,
Rima, can I?
Yes,
Rima,
Partho.
Yeah, first of all, you are asking about.
Sorry for interrupting, sir.
Okay. Arijit, you can carry on, then I will speak.
I just had a question for ma'am.
Yeah.
Yes, Arijit, if you can switch on your camera, I'm not going to judge you for your background. Thank you so much. You look so handsome. I'm happy to see you. Go ahead.
Thank you. Ma'am, I had a question. Correct me if I'm wrong. There was a slide which had compassion or self-compassion.
Self-compassion. Right. Yes.
Ma'am, can you tell me how can a person, especially people who are very self-critic, can increase their self-compassion? How?
Right. Now, Arijit, again, understand that this was a question asked to me, and this is related to this. That's the reason I'm coming to this, that our emotional intelligence is also related to the way we are brought up, the conditioning that has happened around us. How have our parents treated us? How have they handled emotions? We have learned largely from them. And maybe it may be so. It happens in every family. They have their own ways of dealing with things. So maybe so that in your family, people are a little bit more self-critical. They tend to criticize themselves. We all do. And the thing is, if it has been happening for you over the years, good thing, you have realized it, that you are being self-critical. Now, better thing is you have to start working on it. It will not happen in a day, because again, you have been carrying that trait for so many years, right? It's a pattern that every time there's something going wrong, you blame yourself so badly, right? It's a pattern. You wouldn't be able to break it in a day. But the first thing you have done, you have recognized it. The second thing is, next time anything goes wrong, which is not up to your expectation, instead of going on that train of thought that I did horrendously bad and all, catch yourself right there. Catch yourself right there and start writing that key. What are the things that I could do better in the next one? Start writing, and also write what are the things that went well. In spite of many things going wrong, what are the things that went well? Now, this is the key thing you have to do. This is where self-awareness kicks in. Most of the time, we are also not aware that we are having this train of thought that has already left the station, but you have to catch it at the station, right? So that comes with practice. Did that help, Arijit?
Yes, ma'am, that helped.
I just want to add a little bit in Arijit's question.
Please, ma'am, please.
For self-criticism, to deal with the self-criticism, first just let's start thinking that suppose you have a little brother or sister in your home, and the way you deal with her when he or she did some wrong things, you do not the way you console her or him, just start talking the way toward yourself, that yes, I've done it, I did it, but maybe I can do it in a better way in some in next time, or in some other if I get another chance. So I think here also, if somebody experience for long day this kind of problem, one should consult a mental health professional also, because many a times it has been seen that we can read many things from social media, from books, but these things sometimes may not help us. So if you feel that it really creating great disturbance in your life, then you can contact any mental health professional who can just plan a therapy session through therapeutic, in a therapeutic mode. You can have a better life. You can handle your issues in a better way, maybe. Okay, just a little bit from my side.
Thank you for that lovely input, Sanchita. Thank you, ma'am.
I have one question. So is there any exercise for that? Any kind of exercise or meditation will help to treat such kind of psychological problems?
Again, Pratibha, that's a good point you raised. These are all interconnected. Psychological problem, of course, meditation will help. Now, if you understand meditation, I don't know what is your understanding of meditation. My understanding of a meditation as a scientist and a confident personality coach is I close my eyes, I breathe in and I breathe out. Can I keep my head on that, not think of anything else? Can I just focus on breathing? That itself settles your mind down. It sounds so simple, but it's not so easy. Again, over a period of time, it happens. And there is ample research that meditation changes your mind. So of course, going forward, meditation is the way to be. Exercise is again important because your mind and body is connected, but your body and mind is connected as well. You know why? Because see, when you feel depressed or when you feel frustrated, most of the time your body language will be very close. It will be something like this. You will be sitting like this, right? But if you have noticed, when you're very energetic, your body language is open. You know, you are ready to accept anything, right? So our mind controls our body. So when you do exercise, right, too much of your energy goes from your mind away to your body to support that exercise. So you can't be thinking at the same time. You can't be depressed at the same time. That's the reason when you're feeling depressed or frustrated, go on a brisk walk, go running. You will see that you have all of a sudden you're feeling energized. Okay, so exercise always helps. And there's a saying actually which says like body that grooves makes the mind groove. So it's always good. Motion creates emotion. I remember now. Yeah. So it creates the right kind of emotion. Exercise helps.
Ma'am, sir, I also have one question, sir. Yeah.
Yes, Suraj.
Yeah, ma'am. So I have a question regarding decision taking. If you see,
yeah.
Yes. So generally in life, whatever we decide or take decisions, so it do affects our friends, family circle, or organization. So there is always a choice between the emotional decision and the rational decision. So generally we tend to make the emotional decision over the rational, but in the long run we find it difficult to stick on that. So there is a difference. So how we can inculcate to take the rational decision more often, because daily we are finding this thing. Yeah, this situation.
The rational, there is nothing as rational. You're taking that rational decision as well because it's going to make you feel good. There's this feeling good sitting at the bottom of it. That's an emotion, right? So there is a well thought out thing where you combine the rationality and the emotionality, right? Now, when you have to decide, give it some time. And look at it from a space where you can bifurcate the emotion from that. Again, I keep saying that write it down. It's because are you a researcher, Suraj?
I am a professor here in this college.
Oh, okay,
Yeah.
Suraj. May I call you Suraj or I have to call you sir? I'm sorry.
No, it's okay, ma'am. It's okay, ma'am.
Okay.
It's okay, ma'am.
Okay, Suraj. So what you have to do is, when you wrote your thesis, you remember writing your thesis, those bad days, right? Now, when you wrote it versus when you had framed it in your mind, when you had framed the thesis in your mind, right? When did you get more clarity? When you started writing?
Yes, ma'am. When I started writing, I began to develop more insight about that topic.
Exactly. Now, the same thing with decision making. If you have that thought and those things in your head, it's always jumbled up. You don't know where it started and you don't know where it's ending. Write it down, because when you write it down, the clarity flows in. It's like untangling happening. That thread which is very a knot inside your head, it opens up. And when you write it down, you have clarity. That's one of the ways that can help you in decision making. And always, as Vitasta mentioned, think in long terms, not in short term. When you think in long term, that helps you to be emotionally more aware and helps you to think from your executive brain. When we need thinking, I mentioned that slow thinking, right? When decision making happens, if it's career, maybe it requires not fast thinking, sometimes slow thinking. So you want to think from here. So think in long terms and write it down. It's going to give you so much more clarity.
Can
that help?
Yeah. Now can I get my turn? I'm just waiting and waiting.
Yes.
Okay. First of all, when we thought of incorporating such a session, this is the first time I think in Adamus we incorporated such a session inside our conference, and we believed that this would be an icing to the cake of the other speakers actually. But now what I feel at the end of the session is the icing is more yummy than the cake. That's it. That's my point of view or my views of your session. But one thing is, I was just going through your session. I was reading the meals you actually showed to us regarding empathy. When for a student perspective, it's different. For me as a professor, I still like the first mail. So 15 minutes for assignment because we also don't have that much time. So just one liner, 15 minutes for your assignment is enough. That's enough for me. But very nice session, and thank you, Rima.
Thank you,
Madam
Partho.
also. Okay.
Thank you, Partho, for having me. And I hope I can share this on LinkedIn. Is it okay if I take a print screen and share it on LinkedIn?
Sure.
And I just wanted to share some resources that you can use. So these are the resources that you can use.
This time we don't have that much students with us because many exams are going on. Maybe we can have a separate session with you with all our BPharm and diploma guys, so that would be helpful.
Right. Very right.
Okay. Thank you.
Let me just share my screen again. Okay, is my screen visible again?
Yes,
yes, ma'am.
Yes. Okay. And then the reason I want you all to work on your emotional intelligence is okay, we are done with the question and answers. Okay. So I'll just end it on that story again. I had said in the beginning that I failed miserably in that extempore. And then it was in class 11, I decided to participate again in the extempore. And I stood third in that extempore. I was the second runner-up, okay? So it took me 3 years to get past that thing that was like hooked onto my brain that I can't speak in an extempore. Now, why I want you to start working on your EI is because it's the small changes that we make every day. Now, this is mentioned in the book of Atomic Habits by James Clear, that when you start working on improving yourself by 1% every day, just 1%, by the end of the year, say this is February, then by the end of the year, you would improve by 37%. Sometimes we don't see that improvement happening instantly. Again, we are staying in an instant result environment. It won't happen instantly, but you would see that 37% you have improved. And imagine you're starting the next year with that 37% improved version of yourself, and you keep working on you. So the improvement keeps going exponentially. That's the key to improvement. Every leader follows this. Take it from me. Anywhere you go, everybody is working on improving themselves by 1% every day. But if you're not improving yourself, you might be decaying by 2% by the end of the year, which doesn't help anyway. Okay. And if you want to check your EI score, you want to understand have a more better understanding of where you are in terms of EI. This is the website 6seconds.com. You can go and take a free EI test over there. It gives a pretty comprehensive review to start with. Where are you when it comes to the EI thing? Okay, so you can just go and check. It's free. It's anybody can access it. You just have to register. And I'm not promoting it. I'm not getting paid for this, nothing like that. So this is one resource that I have used for myself. That's the reason I am sharing it. And also, this is my LinkedIn profile. And if you want to stay connected, we can stay connected there. And I would be sharing this session on my LinkedIn page. If you can put a comment, that gets me more visibility. That's all I had. Thank you so much.
Thank you so much, madam. I must say that we have just listened to a truly illuminating presentation by Dr. Rima De. Thank you, Dr. De, because your thoughts would remain with us for days and months to come.
Thank you so
help
much.
us to focus on a topic that I mentioned before is becoming significant day by day, emotional intelligence. On a lighter note, it's something that connects your heart and your head. You may have a great skill set, but you are less likely to bring it into a full play if you are not emotionally agile. The professional world has always been highly competitive, and in the post-pandemic uncertain world, that cutthroat competition has become tougher. Only the fittest ones have the license to survive. But who is the fittest? Certainly one who has greater professional skills. And in the post-COVID space, professional skill sets invariably include emotional agility. Life will keep on taking you through several twists and turns. I will, and it will keep on showing you a glass half filled with water, and as we all know, you can call it half full or half empty. Both are true, but on the positive side, one on the positive side and another on the negative side. If you want to turn the negative into positives, no doubt EI or emotional intelligence will guide you. Thank you, Dr. De, for the nice presentation, and my sincere thanks to Adamas University for offering this opportunity to chair such an illuminating session. Thank you so much.
Thank you once again for having me. And on that note, can I have a recording of this session? Can I have access to the recording?
Yes, ma'am. We will send you the recording.
Okay, sure, sure. Okay. Thank you so much for having me. Thank you. Okay. Thank you, Sanchita.
Thank you, ma'am. Thank you, ma'am. Thank you for joining. I think we can relate with your topic very much. Everyone can relate. So 1 hour was not enough for us. So we will meet you again in the next session. So I would like to thank our speaker, Dr. Rima De, for this wonderful and interactive session. And I would like to request Dr. Bitasta Mondal to present the certificate to our speaker. Ma'am, this is a small token of love from our side. So please kindly accept.
I accept it with folded hands over here. Thank you so much. That's a lovely token of appreciation.
Thank you. Thank you, Dr. Bitasta. Next, I would like to thank our chairperson, Dr. Sanchita Ghosh, for being the chairperson in this session and for conducting the show in a smooth manner. And I would like to request Dr. Bitasta to kindly share the certificate of appreciation for our chairperson.
Thank you so much.
Thank you. Thank you, ma'am. Thank you. Thank you all for joining. So with this, we have concluded our session. So with the permission of our chairperson, Dr. Sanchita Ghosh, I would like to end the session. So thank you all for joining. So we will take a short break and we will resume shortly. So please be with us. And till then, please take a short break. So now you can take rest and we will resume shortly. Thank you. Thank you for joining. Thank you all.
Priyanka, are you there? Priyanka? Hello?
Yes, sir. I'm there. I'll be starting shortly.
Yeah. Please start shortly because we are already running late. Okay?
Yes.
Yeah. Thank you.
We are back to the scheduled program after a short break. As Aristotle posited in his rhetorical theory of the three artistic proofs, in order to persuade effectively, one needs to go just beyond the logical facts and include appeal to both emotions and credibility. Good afternoon once again. We have with us Professor Himanshu Shekhar Majhi as a chairperson for the session 3 of day 1 of second international conference organized by School of Medical Sciences, Adamus University, Kolkata, West Bengal. The session will revolve around a topic on challenges of drug powder to solution for formulation development of tablet dosage form by spherical crystallization technique, which will be presented by Dr. Madan Mohan Gupta, Associate Professor, School of Pharmacy, the University of the West Indies, St. Augustine. Dr. Majhi has pursued his B.Pharm, M.Pharm, and PhD in pharmaceutical chemistry from Jadavpur University, Kolkata, West Bengal. He is a dedicated professional having a keen interest for teaching, innovation, and administration in various pharmaceutical colleges. He is presently an Astound Professor of JIS University, West Bengal. I, Priyanka Banerjee, Assistant Professor of School of Medical Sciences, Adamus University, would like to welcome Professor Himanshu Shekhar Majhi, the chairperson for this session. I feel this session will be an interesting and informative one. Now, I would request Dr. Majhi to please head the session forward. Over to you, sir.
Thank you, Priyanka Banerjee, for such a small but beautiful introduction of mine. And I feel privileged to be part of this venture organized by JIS University, organized by Adamus University. Sorry, I am there in the meeting since morning. So they had got a wonderful beginning of the conference entitled "Transforming the Ripples in healthcare research, obstacles, sustenance, and cutting-edge innovations. So after this brainstorming session of emotional intelligence by Dr. Dima De, let us come back to the core subject of pharmaceutical technology. We have with us Professor Dr. Madan Mohan Gupta, who is the Associate Professor in Pharmaceutics, School of Pharmacy, Faculty of Medical Sciences, University of West Indies, St. Augustine, Trinidad and Tobago. Specifically, he will be discussing on the challenges of drug powder to solution for formulation development of tablet dosage form, one of the finest dosage form by spherical crystallization techniques. So to say a few words on Dr. Gupta, he is B.Pharm, M.Pharm in Pharmaceutics, PhD in Pharmaceutical Sciences, MBA and Human Resource Management, PGCUTL from the University of West Indies, St. Augustine, and is presently working as an associate professor in the pharmaceutical at School of Pharmacy, Faculty of Medical Sciences, the University of the West Indies, St. Augustine, Trinidad and Tobago. He worked as a chairman of research committee of the School of Pharmacy, Faculty of Medical Sciences also. He developed a laboratory of pharmaceutical formulation development and design in the School of Pharmacy, Faculty of Medical Sciences, the University of West Indies, St. Augustine. He has worked previously as professor, then principal, and subsequently he is currently working at the School of Pharmacy, Faculty of Medical Sciences. Dr. Gupta is a fellow member of Indian Chemical Society, Indian Association of Pharmaceutical Scientists and Technologists, Association for the Advancement of Biodiversity Sciences, and many more. Dr. Gupta is teaching for the last 20 years to the graduate, postgraduate students at national and international level. And he has supervised more than 16 M.Pharm students and 3 PhD students. Presently, he is supervising 1 PhD student at the University of West Indies. Dr. Gupta has published nearly 80 research papers and review articles in national and international journals of repute. He has filed 3 patents. He has filed 3 patents at the Indian Patents in Government of Indian Patent Office, New Delhi in 2021 itself. He has presented 39 papers in national and international conferences and has organized 8 seminars and workshops as convener and have received about 33 lakhs of Indian rupees grant. He has been awarded as the International Achievement Award in 2nd Science and Technology Award 2014 by EETCRS India. He is the member of AAPS FIP Caribbean Academy of Science. He is a life member of Indian Science Congress Kolkata, APTI, IPTA, IPA, ISTA, ISPOR, and he is the editorial board members of more than 10 national and international journals. So I feel privileged to welcome Dr. Madan Mohan Gupta on this virtual platform at Adams University. Sir, the stage is all yours. Please enlighten us on your lucrative topic on tablets.
Okay, good evening, everyone.
Please, thank you.
Good evening, everyone. Can you hear me?
Yes, sir. You are audible.
Okay, let me share the presentation. 1 minute, please. Okay, very good evening, everyone. I'm Dr. Madan Mohan Gupta, and this is my first time that I'm interacting with you all people. Basically, I'm a person of the pharmaceutics. And for last so many years, I'm working in the solid dosage form designing part. So my today's topic, you can say this is the basically discussion with the young pharmaceutical scientists, or you can say young pharmacy teachers. Other than the novel drug delivery system, what we can do, that is what we can do in the research or in the particular area of the pharmaceutical sciences. That is a principle of the pharmaceutical sciences. Today I will discuss about this, how we can use a spherical crystallization techniques to convert a fine powder into the crystal form. And what is the benefit that I'll discuss in my slides. Because what happened actually, most of time you have seen, even in the market, around 70% market is captured by the tablet dosage forms. So definitely this topic will be useful. Those are doing the research in the fundamental principle part of the pharmacy.
Sir, make it full screen, sir.
My voice is clear?
Yeah, voice is clear, sir. Make the presentation as full screen.
Okay, 1 minute, please. Is okay now?
Yeah, it's fine, sir.
Okay. So like, as I told you that different dosage forms are available: tablet, capsule, injection, liquid dosage forms, semi-solid dosage forms. But out of all the dosage forms, the tablet is a preferred dosage forms. And the reason behind it, because it is easy to use, like patient can easily handle, patient can easily take it. And when you compare with the other dosage forms, like for example, parenteral dosage form or any nano suspension or nano emulsion, its price wise is cheap, that is economic. And you can see in the market that the pharmaceutical world, around 70 to 80% market is captured by the tablet dosage form, whether new drug or whether old drug. But to prepare a tablet, I'm coming on the basic part, then I'll come on the later on the advanced part. To prepare a tablet dosage form, two fundamental things is required. Like you want to prepare any tablet, you have the drug powder. But two fundamental things, that is the first is a good flowability and second is a good compressibility. If any drug powder is having both of the properties, then you can select on the first stage, yes, this powder material is good for the tablet dosage form. But we know that all drug does not possess the granular structure or nature. It means some of the drugs are available in a fine form. If drug is available in fine form, definitely there will be problem on the flowability. Angle of repose definitely will be more than 40. Compressibility will be again one of the issue. So we cannot say that, okay, all drugs is available in the crystal form, I will go for the direct compression method. No. Then you can say, yes, I am having the other option to prepare a fine powder into granules. Yes, I do agree with you. By the help of the granulation technique, either wet granulation or dry granulation, you can convert a fine powder into the granules. I do agree, but what is one of the limitation, you can say, compared to the spherical crystallization, in the wet granulation techniques, definitely there are a lot of steps. If you talk to any pharmaceutical scientist or any formulation scientist, they will say if there are a lot of steps are there, a lot of documentation required, lot of process parameter optimization required, ultimately the cost of the product will increase. This is one of the technique. I'm not saying this is the perfect and only technique, but this is one of the technique. It could be the good technique, less step, and it can provide a more economic product. So by the help of the spherical crystallization, you can convert amorphous and fine powder into the granules. And why you want to convert? As I told you, because you want to increase the flowability. Why we want to increase the flowability? Because it's one of the fundamental characteristics required to prepare a powder into the tablet dosage forms. And these granules not only will use in the tablet formulation, but all these granules will also useful for the capsule during the filling process. I think most of the students, even teachers, know that from the hopper to the die, flowability is one of the important parameters. Otherwise, there will be a lot of, you can say, issue like a weight variation issue or the disintegration. A lot of problem will be there if there is an irregularity in the flowability. But by help of this spherical crystallization, you will see the spherical crystals, and those spherical crystals will be like almost like a round shape. But this spherical crystallization technique is a multiple operation process, including crystallization and agglomerations. And these crystals produce directly agglomerate in the spherical forms. So what is the benefit? Why we want to prepare? Why we are going to use this technique? Yes, we want to increase flowability. We want to change the angle of repose. We want to change the compressibility. We want a good compressibility. We want a good strength of the granules, so we can easily compress a tablet. So by the help of spherical crystals, you will see the good flowability, good compressibility, and good strength. I'll show some data. But now, what are the different stages to prepare a spherical crystallization? That is a fundamental part, that's a basic part of the spherical crystallization. You can see in the picture, to prepare a spherical crystallization, there are different zones. You will not see by your eyes that what is happening, but you know in which step what will be there. Flocculation zone, zero growth zone, fast growth zone, and constant side zone. My dear professionals, these are the 4 steps, but important part is like a design of any dosage forms, whether you are going to design an parental dosage form or liquid dosage form or semi-solid dosage forms. The designing fundamentally divided in 2 parts. One is a process parameter optimization. Another is a product parameter optimization. What does it mean? For example, you want to prepare an emulsion. Okay. For example, you are going to prepare a liquid paraffin emulsion. You know the formula. You are going to add a quantity. Okay. That is you are going to select a quantity, means that is a product parameter optimization. But if you are not selecting proper speed of mixing, proper temperature, proper way of mixing, definitely will not get a good quality of dosage form. Same thing in the spherical crystallization that I'll discuss you in detail. What are the process parameter optimization? In all 4 stages, if you are choosing the right process parameters, I'm talking about only process parameters, then you can get a good quality of spherical crystals. But if you are not selecting the right process parameter, end of the experiment, you will see your crystals are not spherical, maybe weak, or maybe problem in the flowability. So you can see in the picture that the flocculation zone, I will explain you what is this one. Then zero growth zone, then fast growth zone, and constant size zone. In the zero growth zone, you can see the red lines. These are the, basically this is the bridging liquid. What is the bridging liquid? That I'll explain you later slide. And after second stage, you can see these all flocks, that is a particles, they are arranging themselves in a systematic way. And after some time is you have to do some process like a mixing process. Over the period of time, you will see these flocks will be, you can say, they will come together, they will come closer. And after some time, there will be constant growth of your flocks or your particles. Or you can say the fine form will be converted into the good sphericals. You can see here, the flocculation zone means the breathing liquid. I'll explain to you what is the breathing liquid. The breathing liquid displays the solvent from the surface of the particles and form the loose flocks by formation of pendular bridge. You can see here. In the zero growth zone, the loose flocks converted into the tightly packed aggregate. As I told you, you can see here. Initially, there is a more space, but now tightly packed. And the entrapped liquid seeps into the surface of the small flocule. In the fast growth zone, the point where the sufficient amount of breathing liquid has been squeezed out of the surface of the small spherical crystals agglomerate means inside very little amount of breathing liquid will be there. And the final stage, that is a constant side zone. During this process, the arresting of the agglomeration growth means no growth will be after this process. And practically, once you go in the lab and you will start this process, you will see sometimes slightly change in the size of agglomerate means decrease in the size, because they will come closer, they will try to accommodate a less space. Now, what are the basic requirement for the spherical crystal? Three things is requirement. That is a good solvent, bad solvent, and breathing liquid. As I told you in earlier slide, that is a breathing liquid is one of the part, one of the important parameter, one of the important component, which is required to make a bridge between the particles. Good solvent is basically required to make a solution, and bad solvent we call is an anti-solvent, like a good friend and enemy, for example. So three solvent is required, means when you are going to start a process of a spherical crystallization, you have to choose a correct breathing liquid, correct bad solvent, and correct good solvent. And how you can select good and bad solvent? Based on the solubility study. Breathing liquid, generally organic solvents. So three solvent is required, but before this, there are 5 techniques to prepare a spherical crystals: crystalloco-agglomerate, ammonium diffusion, spherical agglomerations, quasi-emulsion solvent system, and neutralization techniques. Out of all techniques, my emphasis will be today especially on the crystalloco-agglomerate. For that, I'm working for last so many years. Okay, this is one of the techniques. I'll explain my crystalloco-agglomerate technique later on. You can see here, you have to mix with the good solvent. Again, listen. First, you have to prepare a drug solution. So you have to select a good solvent where solubility is excellent. Once you are having the good drug solution, then what you have to do, you have to add the anti-solvent. You can say why you are going to use anti-solvent. Yes, because you want to segregate. Then you have to mix it. And see the picture. Because of the breathing liquid, now the both solvent are out and the breathing liquid will make a bridge between the particles. 1 minute. In case of the ammonia diffusion system, so what is the ammonia? We have to use the ammonia water. So what you have to do, you have to prepare a drug solution in an ammonia water. Then we are using the acetone. Acetone is basically organic solvent here. Acetone will diffuse inside. And what acetone will do? acetone will be diffuse out the ammonia water. Ammonia water is good solvent here. And random aggregation will start. And finally, you will see the spherical agglomerates. In case of the quasi-emulsion solvent diffusion system, drug solvent, definitely you have to prepare a drug solution. You have to use the anti-solvent. Then emulsion formation, maybe oil in water, water in oil. Then you can see here, solvent will diffuse out and anti-solvent will diffuse in. Then by the help of the mixing process, crystallization will take place. Yes, this is my technique that I want to explain you. That is a crystal conglomerate technique. This is the, you can say, little more advanced technique. And why it is more advanced, that I'll explain you. First, what you have to do, you have to select a good solvent that is based on the solubility study. You can use the water or alcohol, different solvent. Then this drug solution, you have to pour in a poor solvent. You can see here. And you have to mix it. Same process. Anti-solvent will go inside and good solvent will diffuse out. Then you have to add the bridging liquid. Continuous process of mixing process. During the process of mixing, again I am coming my point, that is a process parameter optimization. And process parameter optimization is very important part. You will see, if you are not going to carefully select the right solvent, right mixing time, right temperature, you will not get spherical crystals. So you have to add a bridging liquid, mix it. Definitely temperature change is one of the important parameter here. Initially, you have to prepare a solution around the 40 degree, that is maybe warm temperature. Suddenly, you have to decrease the temperature. Why? Because you want to make a crystals. You can see here, this is one of the drug, that is a valsartan. One of my students did the work on this one. Prepare a solution of valsartan in a good solvent. We select this NN dimethyl formide based on the ternary phase diagram. And you can see here temperature is 65 degree centigrade. Then what we did actually, we add the talc powder. You can say, why we are adding the talc powder? Yes. Sometimes what happens, when you are mixing, because we are going to prepare a spherical crystal, you can see we are going to reduce the temperature suddenly. There is a possibility that particle can come together very fast and they can make a big particles. That is a clumps formation that we don't want. So this talc powder will use as a lubricant here, like in case of tablet. So this solution, that is a solution in a good solvent, which is having temperature around 65 degree temperature. Sometimes depend on the solubility, you can add around 45 or 50 degree. But this particular valsartan, we use a 65 degree temperature. And you can see, we put this solution in a 5 degree temperature. Where? In a bad solvent, in an anti-solvent, in a poor solvent. For valsartan, water was an anti-solvent. That is a bad solvent, or you can say anti-solvent. Two things is here. First thing is, we are adding a solution that is a good solvent in a bad solvent. And second thing is a temperature. And you know that because of the temperature change, crystal formation will take place. After addition of this one, we add the bridging liquid, that is a dichloromethane, that is a DCM, and we start a mixing process at optimized speed, that I'll explain you what we did actually, and as well as the temperature. Temperature and speed is a process parameter. Addition of bridging liquid, how you are adding. Listen, selection of bridging liquid is a product parameter. Selection of bridging liquid is a product parameter optimization. What type of the bridging liquid you are selecting. But how you are adding a bridging liquid is a process parameters. Difference. Like whether you are going to add at one time or drop wise, that is a process parameter. But you are selecting the acetone or dichloromethane, that is a product parameter optimization. Addition of talc powder, you already selected talc powder, but how you are adding, that is a process parameter. How you are mixing, what is your speed of mixing, is a process parameter. What is the temperature, that is a process parameter. Okay, so after this one, mixing and temperature, both things. Over the period of time, you will see, I'll explain you different by the base of the data. You will see that the formation of the spherical crystals. Then what you have to do, sometime you have to keep this solution overnight, or sometime if you think that is okay, because once you go for the experiment, you will see, you have to filter out. Once filtration process is over, you can keep all this spherical crystal on the room temperature in a desiccator between 25 to 30 degrees, that is a room temperature. Yes, that is a process development, that is a process parameter optimization. Because in the spherical crystals, the significant part is a process parameter optimization. You can select a good bridging liquid, but if you are not adding in a proper way, you will not get the spherical crystals. So what is the measure? There are a lot of process parameter optimization, but this major one is agitation speed, agitation time, temperature, and how you are adding the bridging liquid. So first is, this is a product parameter optimization. We use a hexane, we use a toluene, we use a dichloromethane, benzene, chloroform, and isopropyl alcohol. Out of all these, what we found actually, dichloromethane, I am talking about the spheric, especially for the valsartan. Okay. Dichloromethane was good to prepare a spherical crystal, but hexane, toluene, benzene, chloroform, all these organic solvent, there were clump formation that we don't want. Because size of the spherical crystal is also important parameter to prepare a spherical crystals. And in case of the IP, that is isopropyl alcohol, what we found, there is no crystals, clear solution. So it was a product parameter. Now see, this is the process parameter optimization. You can say what is meaning? Yes, you can see here 300, 400, 600, 800. For your experiment purpose, you can start from the 100, 200, 300, 400, 500, 1000. But based on the literature, actually, we select the 4 different speed: 300, 400, 600, 800. At the speed of 300, what we saw actually, the clump formation. Why? Because speed was not sufficient, and because of that, the particle, they came together, they form a clump. At 400, again same, because a little bit changed, a small compact. 800, irregular shape. Why? Because speed was very high, and because of the, you can say the mixing effect, or because of chopping process, because of high speed, the particles were there, but irregular shape that we don't want. We want a spherical crystal. Why? Because ultimate aim is increase of flowability. If flowability is not increased, then what is the use? Again, problem will be angle of repose of flowability. There is a possibility if you are going to prepare a tablet, weight variation problem, lot of problems. But at 600, what we found? Yes, spherical crystals with a good shape and size. For your experiment purpose, you can start from the 100, 200, 300, depend on your drug. But you have to optimize the speed. You cannot say randomly I will select the 500. No. Second, how much time you are going to mix. You can see here 5 minutes, 10 minutes, 15 minutes, 20 minutes, 25 minutes. This is a speed basically. 1 minute. This is a speed where you are mixing here. Yes. 5, 10, 15, 20, and 25. When we use the 5 minute only, that is small amount of time in incomplete agglomerate because time was not sufficient. When we use the more time, 15, 25 or 20 minutes, you can see in a 15 minute clump formation, 20 minute clump and broken spherical crystal. Why? Because you are mixing over the requirement. For example, you want to polish something or you want to make a shape of something, you are using more time of the, you can say fracture, or you can say some polish or something mixing. Definitely you will see the structure will not be proper. Same thing in case of the spherical crystal. If you mix for the more time, whatever spherical crystal you are having, now they all will be like a broken shape or maybe irregular shape. If you use a more time, 25 minute broken spherical crystal, or if you go for like 30 minutes or 40 minutes, 50 minutes, you will see spherical crystal but different shape, irregular shape, some is fine powder also. So you have to optimize. Same thing like, for example, I think everybody remember, those are working in the tablet dosage forms. In the tablet dosage form, one of the steps is you have to mix like sometime magnesium stearate or talc powder, lubricant and glidant. In process, tablet formation process, when you are using the talc powder or magnesium stearate or carboxyl silica, all these things. If you are blending, I'm using the word blending, not a mixing word. If you are blending more time, talc or magnesium stearate with the granules, there is a possibility of, again I'm repeating, there is a possibility of charge development surrounding the particles. And because of that, you will see the rate of drug release will be different. You can say, how is it possible? I use the good quality of talc powder or good quality of magnesium stearate. Everything was okay. My product parameter was excellent. It was because you did not select right process parameter, means you mix more time over the time. Same thing in spherical crystals. You can say, if I will use the more time of mixing, my crystal will be good, excellent. No, because after 10 minutes, because now spherical crystals are there. But if you mix more time, now what will start? The clump formation, then breaking process, and then fine powder process that we don't want. So that is an optimized speed of mixing. Temperature. You can say my initial temperature of solution is a 50. So if I'm going to use a 25 degree or 30, that is okay. Yes, that is okay. Depend on your drug. Because you want to change the temperature. I do agree with you. But like in case of our valsartan, or generally, like in my case, we use a 5 degree plus minus 2 degree spherical crystals, but at 40 degree or 25 degree or 30 degree, no agglomeration, no crystals, no nothing. It means temperature, that is a process parameter, is also one of the important parameter. You are selecting the right speed, good. You are selecting right bridging liquid, good. But if you are not selecting the right temperature, you will not get a good quality of the spherical crystals. Now, another process parameters. It is a very small, sometimes, okay, what is the problem if I'm going to add a whole amount one time? What is the problem? Yes, you can see. If you are adding your entire amount of bridging liquid one time, irregular shape of agglomerates, size will not be good, irregular shape, flowability will be problem. So what you have to do, you have to add a drop wise. One drop, then mix it. 1 drop, then mix it. You can say, what is the problem? In only 1 ml amount or 2 ml amount, I can at one time, because the amount where I'm going to add is a 50 ml or 100 ml. Yes, 100 as compared to 100 ml, it is 1 ml is 1%. But no, if you add one time, there's a possibility of the irregular shape agglomerate. Why? Because bridging liquid will accumulate only one place. You have to add drop wise. So another process parameters. You can see here, this is one of our drug, valsartan. Before the spherical crystals, angle of repose was 42 degrees. That is you can say very poor flow drug. You can see other parameters, that is a car index, Hausner index, step density, bulk density, all are not good. You can say these parameters say that you cannot prepare a tablet without making a granules or crystals. But after the spherical crystallization process, you can see here there are 3 batches. We prepare a lot of batches, but I'm mentioning here only 3 batches. Like in second batch, 25 degrees angle of repose means excellent flowability. You can see here bulk density, car index is 4.21, excellent compressibility. Means now your powder is ready to prepare a tablet dosage form in a crystal formation or a spherical form. You can see here the picture, that is a SEM of the valsartan spherical crystals. Pure drug was looks like a lumps or something like a fine powder. But here you can see agglomerate is a excellent, or you can say spherical crystals, round shaped crystals, excellent crystals. So this is my small presentation, and I would like to come to the conclusion part. The spherical crystal technique can be troubleshoot the flow problem of the fine drug powders, because this is one of the big issue for the pharmaceutical scientists. Those are working in the tablet section, because they are having only 2 options, whether they have to go for the granulation techniques or they have to reject it. By this techniques, they can prepare a powder into the spherical crystals, because there are less steps as compared to the wet granulation techniques. Definitely less documentation process, less optimization process, and definitely final, the cost of product will be less, or you can say economic process. I would like to acknowledge my University of West Indies and one of my students, Adnan Chatterjee, who did this work also. So thank you, everyone. I would like to.
Thank you, Dr. Gupta, for your nice and elaborate presentations. So the house is now open for question answer session. I would like to request our dear participants to please unmute yourself and ask any questions if you are having on this subjects of crystal co-agglomerations. Priyanka, you can also assist.
Yes, sir. Sure. Okay, it would be better that if all the participants preferably just down your questions, it would be easier for Manji sir as well as me to get back to the questions.
Yeah, this is a topic which can be related to every person, say starting say B Pharmacy first years, diploma pharmacy, till the masters or PhD students of pharmaceutical fields, because this is such an interesting topic Professor Gupta has discussed today. So hopefully there will be many questions from the participants. Participants, please, if you have any questions, you are free to ask.
Sir, I have a particular question, but I am very sorry that I am unable to switch on my video. I was trying since a long time. Shall I ask the question to Dr. Gupta?
Yes, yes, please.
Yeah. Thank you, sir. Sir, I was having a confusion about one thing that you have spoken a brilliant, or you have given a vivid description about the topic which was about this session. So this was about the spherical crystallization technique. So what light do you want to throw on co-crystallization technique? Is it the similar one, or do you think that it's a further advanced step to this spherical crystallization technique? Can you please brief me about this?
Yeah, the spherical crystallization is a broad topic basically. And how to prepare a spherical crystals, out of that one technique is a crystallo-coagglomerate techniques. Dr. Banerjee, did you understand what I'm saying? The spherical crystallization is a broad topic. There's a 5 techniques: ammonia diffusion, neutralization techniques, like the same is a crystallo-coagglomerate techniques. This crystallo-coagglomerate technique is one of the advancement, like I mentioned in my PowerPoint. We are using the talc powder. We are optimizing a lot of parameter. Because of addition of the talc powder, it is a crystallo-coagglomerate. We are going to agglomerate the crystals to make a better spherical crystals.
Right, sir. Right, I got it. Thank you for the same. I have already got the answer which was being required for this session. Maji sir, would you like to take some of the questions? Are there any questions? Sure, sir.
Sir, with your permission, I am having a small query.
Yeah, good.
It's question. Sir, I was going through your crystal agglomerations techniques for valsartan, where you are preparing the smaller size particles. So what I believe is that temperature as well as the stirring speed, both are required for proper crystal growth. So you have mentioned regarding the temperature, so from 65 to 5 degrees. One good solvent which is at 65 degrees centigrade, then you have incorporated one bad solvent which is at 5 degrees centigrade. And at the same time, the stirring of the mixture you have mentioned, sir. What could be the RPM or any optimum RPM that needs to be maintained for the preparation of crystals of valsartan?
So as I mentioned in my PowerPoint, 300 was a RPM that we selected for this prepared spherical crystal. We use a 200, we use a 300, we use a 600, we use a 800. Out of those, actually, let me share you my presentation.
So this optimization has been done by you?
Yes, yes. Actually, we use initially 100, then 200, then 300, 400, different speed, and based on the literature, we found this is okay. Then we do the experiment in our lab, then we found this is a okay, that is a speed.
Okay, sir. My second query on this is that whether this is true for any other drugs that specifically mentioned for valsartan. So
no, you can use for any of the drug, which is a fine powder. You have to optimize the process parameter. You can go ahead, because we also
with 300 RPM, right?
Yeah, speed you have to optimize. Like we did the work on the aceclofenic, we did work on the QT10 fumarate, we did the valsartan. So any of the drug which is fine powder having the flowability problem or compressibility problem, and you want to prepare a tablet dosage from, you can use this techniques. Only important thing is you have to optimize the process parameter. Right. It could be possible in your case, maybe 500 speed is okay, like sometimes 300 is okay, sometimes 700, but you have to optimize, because if you are not optimizing speed, finally you will not get a good quality of the spherical crystals.
So both the variables like temperature and RPM both needs to be optimized in situ.
Yes.
Yeah. So there is no standard, say, norms or something like that.
No, sir.
And at the end of the crystal growth, sir, how you are analyzing that the proper crystals has been prepared?
Once your crystallization process over, there are a lot of analytical technique, like you can go for the DSC, you can go for the XRD, you can go for the SEM. These are different techniques. You can find that okay, shape is okay.
Okay. So what I can understand, sir, it's a new techniques also to me, is a new form of this particle size reduction, what should I say, to improve the bioavailability in the tablet dosage form. I'm done. If there are any more questions from our participants, or else we can. Anyone? You can please unmute. Dr. Gupta is there with us for a few more minutes. Yeah, okay. Okay, then, sir, I don't find any further questions in this presentation, but what I believe, or we believe, that this presentation will be an eye opener, and it's a new technique for this particle size reductions and tablet dosage form formulations development. And on behalf of ourselves, on behalf of Adams University, we would all like to thank you for enlightening us with your lucid lecture on challenges of drug powder to solution for formulation development of tablet dosage form by spherical co-crystallizations. I personally have learned a lot. Thank you. Thanks a lot, sir.
Thank you, sir.
Over to Priyanka.
Thank you, sir. Thank you, Dr. Gupta, for such an insightful session. I would now request Ms. Pratibha Gupta, Assistant Professor of Adams University, to present a gratitude certificate to our respected speaker, Dr. Modan Mohan Gupta, Associate Professor of School of Pharmacy, the University of the West Indies, St. Augustine, for such an eventful session. Sir, this is a small token of appreciation you can consider from the Adams University team, and it's all for you. We would request you for further session, if it would be a privilege for us that you could come down to the college, and after this scenario, we will get to hear from you soon. Thank you, sir.
Thank you so much.
Thank you, sir. Thanks, sir.
Thank you, sir. I would also like to acknowledge the contribution of the chairperson of this session, namely Dr. Himanshu Shekhar Majhi. He is the professor of GIS University, as I have already mentioned. And I would again request Ms. Pratibha Gupta to share the appreciation certificate for sir as well. Sir, please.
Yeah, thank you, but I believe this is not required for me in presence of Dr. Gupta, but I feel honored. Thank you. Thanks,
thank you, sir. Thank you. Thank you, everyone. So on behalf of School of Medical Sciences, Adams University, I would request the permission of our chairperson of this session so that I can end this session now. Sir, would you please permit me to end this session?
Yes, please. Go ahead.
Thank you, sir. And would like to inform all the participants to gather for another last session of today for the second international conference from 7:30 p.m. IST sharp. Thank you.
Thank you so much.
Yeah, I am leaving. Thank you. With the permission of the host.